So good morning, everybody, and welcome to the Highlighting Organic Chemistry in Portugal, part of the series organised by the Division of Organic Chemistry in EuChemS. So it's a great pleasure to introduce Professor Arthur Silva, who will give a brief presentation about the importance of organic chemistry in Portugal. Over to you, Arthur.
Good morning to all of you. Welcome to this webinar about highlighting the organic chemistry in Portugal. And I thank also all the attendees and all the panelists, and especially Pat for organizing these events with all the organic chemistry divisions of EuChemS. So I can start saying that where we can have research in organic chemistry is in main universities in mainland, that it's highlighting here in red. It's University of Minho, University of Porto, University of Trás-os-Montes de Douro. Then we go for we try to see my pointer, please. Yes, University of Aveiro, University of Coimbra, University of Beira Interior. In Lisbon, we have two: University of Lisbon, New University of Lisbon, University of Évora, Algarve, and in Iceland, Madeira and Azores. And of course, that in these groups that are bigger, some are smaller groups doing some research, but we can have real organic chems and other natural products, and we can put this in framework of the organic chemistry. And besides that in university, the research that are doing this university, in some polytechnic institutes, there is also some researchers doing some working in natural products, that I can say. I can give a small overview about the organization of the research in Portugal. We are organizing research centers. Some of these research centers are composed only by chemists, and other are interdisciplinary. It means that we have in these research centers not only chemists, but biologists, material science, and so on. These research centers where we carry out the organic chemistry research, we can have academic staff that how we start in the 70s and 80s and so. But nowadays there is also researchers we have a career of full-time researchers, and nowadays we have some with contracts with the projects, but there is also some that have a tenure position for all the life. And of course, we have PhD students that can bear individual grants from the Foundation for Science and Technology, and also those that are involved in projects. All of us are organized in the Portuguese Chemical Society. We are associates, and we have two main divisions: the organic chemists and the division of medicinal and biological chemists, where the organic chemists are associated. And of course, we are members of the EuChemS and also of the organic chemistry division of the European Chemical Society, for sure. Only a small evolution of the organic chemistry. In the 70s and the 80s, we had at that time poor infrastructure, and the organic chemists worked in collaboration mainly with UK and USA colleagues, and with a lot of our colleagues, our former bosses, have done the PhD in these countries, in these research groups, and they have some summer internships. When they have a break in Portugal from the teaching duties, they went to these countries to do some summer internships, and also to carry out the structural characterization, because at that time we almost didn't have any equipment or big equipment. And in the 90s there was a project, let's say, a program where we had the opportunity to have naval infrastructure and equipments, and that in these 90s we start to have NMR facilities, MS facilities, and then there was a big evolution. But it was in 90s when we have this, and also the collaboration were extended to other countries. before it was not UK, it was not only UK and USA, but was mainly USA. But after the 90s, we have opened and extended this collaboration all over the world, but mainly in Europe. And in that time also, we start to have grants for PhD students, grants from the Foundation for Science and Technology. The name at that time it was not like this, but it was the foundation that have organized all these individual grants assigned or attributed to the PhD students in Portugal. And then we start to have also postdoc fellows. That with the postdoc fellows, there was a great advance in research and mainly in all the research in Portugal, but also in organic chems. And from in the 21st century, we start to have these full-time researchers, this career, this alternative career to the academic staff that also have a great impact in the research, all the research in Portugal, but also in the organic chemistry research. Yes, this is only to show a little bit the evolution of the documents. Of course, that I tried to put it here, the organic chemist, but when I have done this search, I put the organic chemist, but then I see that is not only the organic chemists, but also other colleagues that have done research that can be associated with organic chems. But this show a little bit the evolution how we have in terms of science. It means that until the 19th, you see, it was residual, the publication that we have, and now we have a big impact, and now we are more or less in the stand-up with a good number for the number of research that we have in Portugal. So it was a brief overview of the university, of the research, in the organic chemistry research, and we can also say that the colleagues that start the organic chemistry research in Portugal nowadays are retired, and the presenters that we have today is the second generation, can I say, and now we learn, we start to have these facilities, and now we are carrying out the research in Portugal. All of the presenters have and has important duties with the organic chemistry division and also with Portuguese Chemical Society, and I can show a little bit what we'll have. We will start by Teresa from the University of Coimbra with the main with the organic chemistry and the discovery and the disclose of biological active compounds. Then we have Augusto Tomé from the University of Aveiro with a specific group of organic compounds with synthesis and biological application. Then we have chemical biology with Pedro Góes from the University of Lisbon. And then we will have Vitor Freitas with a group of compounds that is studied for a long time from the University of Porto. Then we moved toward the University of Lisbon with Paula Branco, that is explored an alkaloid, it show the synthesis and one alkaloid and related compounds. Then we have Anthony Burke, that it was in format before in the University of Évora, and now it is in the University of Coimbra, doing or speaking about the synthesis and using catalytic multi-component reactions. And we will finish this with synthetic transformation of biomass using under flow conditions. So this will be what we'll have in the morning, and I thank all the colleagues to be with us and present some of the research that they are doing at their research centers and universities. And I thank you also for listening to me to give this overview of the organic chems, organic chems in Portugal. But
Arthur, thank you very much. Very interesting and very informative. So if you could please stop sharing your screen, and we can ask Teresa if she could please start sharing hers. Now, Arthur, you're chairing this, so I'll ask you to please introduce each of the speakers.
Yeah, well, Teresa Pinhamelo is professor at the University of Coimbra. She is a long historian in the organic chemistry division, looking to the do the synthetic organic chems division for I can say some heterocyclic chems, but also some using some cycle addition reaction and so. So Teresa, the floor is yours.
Okay, thank you. Thank you for the invitation to participate in this webinar. So the topic of my talk is organic chemistry and discovery of new biologically active compounds. What I would like to talk today is about two research topics, which are anchored in organic chemistry, but with the focus on medicinal chemistry applications. So novel spirobeta-lactams with broad spectrum antimicrobial activity and novel tetrapyrrolic macrocycles for photodynamic therapy and diagnostics of cancer. So let's start with these novel spirobeta-lactams. So the starting idea was to prepare beta-lactams, spirobeta-lactams, having the core structure of penicillins, which are this bicyclic system, and adding additional structure feature, which is a chiral spiral carbon, which normally is associated with the biological activity of natural spiral cycle compounds. So our synthetic strategy was to start to explore the reactivity of diazopenicillinates and alkylidine penicillinates, and these are obtained from 6-aminopenicillinic acid. This is a commercially available compound which is obtained from the hydrolyzed side chain of penicillins and is chiral. So it allows us to carry out the stereoselective reactions. So we were able to obtain a range of these spirolactams, which are in fact penicillin analogs, having this additional ring that can be a carbocycle or a heterocycle compound. So this project started as a PhD, in fact, a PhD project at Brno Santos, and from this initial study, we built a library of compounds. And from these compounds, we identified these three derivatives showing high anti-HIV, anti-plasmodial activity. As you can see here, we have derivatives having this cyclopentane ring, but also this pyrazole ring. At this stage, we established a collaboration with Nuno Taveira from IMED, University of Lisbon, which is an expert in HIV infections and other infectious diseases, and Miguel Provencio, which carry out research on malaria. So one of our best performing compounds, which in fact became our lead compound, show high activity, nanomolar activity against HIV-1, HIV-2, and also against Plasmodium, 2 stages of infection, liver and in blood stages of infection of Plasmodium. And so we, of course, carried out several structural modulations. And in this case, we started with these 2 types of spirolactams, the ones having the cyclopentane ring and pyrazole ring, and you obtain a pool of compounds, 23 new spirolactams. And from this study, we identified 2 additional compounds very active, also with this dual activity. As you can see, both compounds have this cyclopentane ring. So we then focus on this type of derivatives. We extended our study to further modulations, and we obtained, in fact, we obtained 8 spirolactams with dual activity, very active. And from these, 3 of them were, in fact, very active against these 2 pathogens. Of course, these allow us to gather a lot of structural activity relationships, and now we can design new modulations, rational design of new derivatives. In fact, the cyclopentane are the more promising derivatives, and these are obtained from these 3+2 annulation reactions. We start with alkylidine, the reaction with alanoids in presence of phosphines generates these deuterionic intermediates, which can lead to 2 radioisomers, depending on the initial attack of this alpha carbon or of this gamma lactam. this gamma position, this gamma carbon. But in fact, the relevant compound regarding biological activity is this gamma radiation. And we obtain these compounds as chiral compounds, seroselectively, because these alkylides have this butterfly-like conformation. This is one X-ray structure of one of alkylidine derivatives, and the incoming reagents will approach this less hindered alpha phase. So in fact, it is the seroselective reaction, transformation, that allows us to obtain chiral compounds in pure form. More recently, we were able to develop conditions, continuous flow conditions to prepare our lead compound. And in fact, we observed that the production rate is significantly higher than conditions under batch. Of course, this will allow us to study easily scale-up studies. Okay. So we are very excited with these results because with this dual activity, if you consider HIV in 2022, there were 39 million living with HIV, and malaria also affects millions of people. And there is some geography overlap, in particular in some parts of Africa about regarding these two diseases, infectious diseases, which leads to the spread of both diseases. So substantial number of co-infections, malaria enhances the HIV replication initial levels, and HIV infections increase the risk of malaria. So as I mentioned in this, the title of my talk, there are partners in crime. And there is also this problem, malaria and HIV are treated with different drugs, so there are potential unwanted drug-drug interactions. So having one molecule with this dual activity, it is particularly interesting. So, of course, we look into the mechanism of action, trying to understand the action of our lead compound. And one experiment that we carried out is this time of addition experiment. Here, what we want to know is how long can we postpone the addition of our compound without losing activity? And you compare it with the experiments with drugs that are known to target the different steps of the HIV replication cycle. Here, our compound is here in black, and you see that only after 18 hours, there is a decrease in activity, which means that our compound could act in the last stage, last step of the replication cycle, which is maturation and release of the virus. This could be that it will be a protease inhibitor. However, we show that the compound does not show any protease inhibitor activity. So our conclusion was the compound acts on the cell rather than on the virus. So the fact that these results of this experiment, together with the fact that it is active against two different organisms, in fact suggests that the compound targets those cells. And what we believe is that it triggers the immune response, releasing interferons or other proteins that from the immune system that will inhibit the replication of the virus. But what this means that the compound probably will be active against other virus, potential activity against other virus. So we extended our study to other viruses, and in fact, the compound inhibits the replication in influenza A and influenza B with very low C50 virus. And these lower viral respiratory infections are in fact associated with potential pandemic situation. Of course, you all know the results of this pandemic, COVID pandemic. The lower respiratory infections are responsible, the fourth cause of death globally, and the pandemic, COVID pandemic was an example of the threat to public health. So we also explore the activity against SARS-CoV-2, and compound also was particularly active. So also no relevant cellular toxicity. no antibacterial or antifungal activity, stable immunoplasma. So we think that we can develop this compound as a new broad spectrum antiviral. At this stage, we have several compounds. We prepared 60 compounds directly derived from our lead compound. And at this stage, we identify already 19 compounds with IC50 below 200 nanomolar, and 9 of them with IC50 below 50 nanomolar. So in fact, you have several compounds at this stage which are very promising as broad spectrum antiviral compounds. This work was, in fact, carried out by Américo Alves and Nuno Alves as their PhD project, and Nuno Alves was the spokesman of this award, born from Knowledge, which was an initiative of the National Innovation Agency, and also the team recently received this award as the Gilead Award, which is an incentive to pursue this line of research. So the second topic is novel tetrapyrrolic macrocycles for photodynamic therapy and diagnostics of cancer. And I'd like to focus in two families of compounds. Ring-fused currents, which is chemistry is based on an unprecedented +2 cycle additions of porphyrins with diazophovenometides, and corals, oximes and diazone functionalized corals, which since was developed by exploring the reactivity of nitrose and azo alkanes with dipyromethanes. So let's start with this novel type of ring-fused currents. So, which, as I mentioned, is based on this 8+2 cycle addition of diazophovenometides to build this azocyclic ring. In fact, the chemistry of azo and diazophovenometides has been a research topic for several years in our group. But today I would like to focus only on reactivity to prepare these novel tetrapyrrolic macrocycles. And the work started, this research topic started as a PhD project of Nelson Pereira. So, as you know, PDT relies on a photosensitizer activation, open light irradiation, leading to reactive oxygen species. And these reactive oxygen species are responsible for the cytotoxic effects. So our goal has been to develop more efficient photosensitizers in order to obtain more efficient PDT treatment. And we were able to demonstrate that porphyrin can act as 2p components in this 8+2 cycle addition. And you obtain a new type of current derivatives. Stable currents, and we can easily modulate the structure of these compounds. For example, we can replace the phenyl group by other aryl groups by starting from different porphyrins. And you can also carry out functional group transformation of these ester groups, for example, the reduction to the diol derivative, a selective reduction of one of the ester groups, or the hydrolysis of the ester groups to the corresponding carboxylic acid. All these derivatives show absorbance within the phototherapeutic window, which is a requirement for a good photosensitizer in order to reach deeper the target tissues. So just our initial results, we started, I'm just going to show selected results of the compounds derived from TPP, tetraphenylporphyrin. So what you observe is that we started to study the activity against melanoma and observed that the diol derivative show nanomolar activity, very low IC50 values, much more active than the corresponding ester current derivative. And you were able to explain easily these results, consider that the diol derivative current 2 presents solar uptake values 10 times higher. Of course, we extended we prepared a range of derivative, and as I mentioned, this included the acid derivative and this monoalcohol, and compare it with the diol derivative. Initially, as I mentioned, we started determining the activity against melanoma, but then we also studied the activity against blood cancer cells, esophageal cancer cells. So all these derivatives, the diol, the diacid, monoalcohol, they all have the absorption within the phototherapeutic window. And as you can see here, the Corin 5 is the diol derivative. It is particularly active against this cell line study, very low IC50 nanomolar activity. The diacid is particularly active also against melanoma, with moderate activity against the other cell lines, but still very interesting results regarding melanoma. And then the monoalcohol is less active, although against melanoma there is also the IC50 is quite low. We normally study the photophysics and photochemical property of these derivatives. And in this case, you can see here the single oxygen formation continuum, which does not correspond to the phototoxicity observed, because the acid derivative is the one showing the higher single oxygen continuum. So having two ester or one ester group leads to higher phi delta values. It's not in agreement with the observed activity. However, it correlates well with the generalization, the uptake values. So if you can see here that with two esters or one ester, we have low uptake values. The acid derivative shows intermediate values, and the diol derivative is the one showing higher uptake values, which in fact is the more active photosensitizer. We studied different modulations, and in this case, what I would like to show is the diphenyl derivatives. So the strategy rationale was that the presence of two phenyl groups instead of four should reduce lipophilicity of compounds, and this should lead to higher uptake values. And in fact, we studied again these three types of cancer cells. And in this case, even the ester derivative is very active, very low IC50 values below 50 nanomolar. But of course, the diol derivative are even more active. For example, melanoma showing IC50 values below 5 nanomolar. And for one of these derivatives, we carried out these in vivo sheet embryo studies. So in this model, the tumoral cells are grafted in this upper membrane, and then we carried out the PDT treatment with our compound and can follow the results. And what you observe is a tumor regression of around 33% with a single PDT treatment. So this is considered a proof of concept in vivo study and confirmed the potential of this derivative as PDT agent. Of course, we expanded also the chemistry to other derivatives, in particular these pentafluor phenyl porphyrin derivatives. Having these pentafluor phenyl groups allow us to introduce different groups at pair positions, exploring aromatic nucleophilic substitution. So we prepared the 8+2 cycloadduct. We introduced these amino peg chains, these short chains. We also carried out the reduction of the initial adduct. In this case, there was also the removal of the fluorine atom of these positions. And we combined the introduction of these amino peg groups with diol moiety. So we, in fact, we modulate the polarity of the compounds. And we studied activity against melanoma and esophageal cancer cells. As expected, the ester was not very active. Then when we introduce these side chains, we have already a significant impact in activity. But it was interesting to observe that the reduction of the ester groups have an even higher impact, and we obtain very low IC50 values. And if you combine the diol moiety with these side chains, the compound is even more active. So against melanoma, IC50 of 13 nanomolar, and against esophageal cancer cells, 27 nanomolar. So in fact, we were able to modulate significantly the properties of these compounds. In fact, what we achieved was the modulation of the photophysical properties, because there was a decrease on the fluorescence quantum wheel, and an increase in singlet oxygen formation continue, and also improve the cellular uptake values. So we end up with these very active PDT agents. More recently, I'm just going to mention some highlights of this study. We prepare Foscar-derived compounds. Foscar is a clinically approved chlorine, and what you were able to demonstrate is that preparing this ring-fused system, you obtain compounds with higher photostability, which were safer in the dark, and with similar or improved phototherapeutic index. So it is a strategy instead, in fact, to modulate the property of these chlorine derivatives. So what I would like now to talk about is diagnostics, imaging and therapy in a single system. And what you observe is that introducing platinum in this system, we obtain, in fact, a compound that can act as diagnostic agents. The chemistry is similar, but in this case, we start with this platinum complex, the porphyrin platinum complex, carry out that plus 2 cycle addition, obtain the corresponding chlorine derivative, and then we can carry out the modulation of the ester groups, the reduction to obtain the diol, or the hydrolysis to obtain the diacid. And of course, we studied, I'm just showing again some selected results, so the compounds derived from TPP. And you were able to show that the platinum derivatives can also act as PDT agents. Particularly against melanoma, we obtain very low IC50 values, although the diol derivative was more active than the diacid derivative, in particular against melanoma. So they can act also as PDT agents. And what was interesting to observe for the diol derivative is that they show simultaneous fluorescence and fluorescence at room temperature. So they can be used in imaging, and they can act as oxygen sensor. So here I'm showing the emission in the absence of oxygen and in presence of oxygen. So it can act as oxygen sensor. And as I mentioned, it can act as PDT agent. So in fact, the compound is a diagnostic agent. And we were able to validate this conclusion, carrying out this in vivo study. This time we use a melanoma mouse model with the tumor grafted in this axillary region. And what I'm showing here is imaging with our compound. And you can see the compound accumulates in the tumor. And also we could observe with a single PDT treatment, a significant reduction in the tumor volume. And we confirmed that the compound accumulates selectively in the tumor by carrying out this biodistribution study, although there is some accumulation in the skin. It's an issue that we are trying to address now. So the final topic, the second family of tetrapyrrolic compounds are chorols. And these were developed, the synthesis was developed by exploring the chemistry of nitrose and azo alkenes. The chemistry of nitrose and azo alkenes has been also a research topic for some time in our group. And we focus mainly in the functionalization of 5-member heterocycles, but also in the developing a synthetic routes to heterocycles. I'm just going to mention one of the methodologies that we developed, which is the synthesis of bis heterocycle methanes, in particular dipyrometanes. So the strategy is based on this reactivity of heterodienes, as I mentioned. We start with oximes or hydrazone, dehalogenated oximes or hydrozones, and we carry the elimination reaction, dihydroelimination reaction, giving this heterodiene, which participates in a heterodiazole reaction with pyrrole to give this cycloadate. Opening of this 6-member ring leads to this functionalized pyrrole. We also demonstrate that in some cases, mechanism pathway is different and can involve conjugated addition followed by rearomatization of the pyrrole. Nevertheless, we obtain this functionalized pyrrole with this side chain, then can undergo another elimination to generate a second heterodiene, which reacts with a second molecule of pyrrole to give the dipyron 10. So in fact, we develop, it is a one-pot synths of giving this dipyron 10 functionalized with oximes or hydrazines. So we thought that replacing pyrrole by dipyron 10 will find a way to prepare tetrapyrrolic compounds. And this was the base of the methodology to obtain the Courant derivatives, exploring the reactivity of these nitrose or alkanes to add dipyron 10. We started with oximes. These are now the PhD project of João Branch, and we were able to in fact show that the reaction starting with dehalogenated oximes, reacting with dipyron 10, we obtain these tetrapyrrolic compounds, these Villen derivatives, and then we can carry out an oxidative macrocyclization to obtain the corresponding corals. And we obtain a range of derivatives, which included different aryl groups at meso positions, but also different groups directly attached to the oxime group. Can be aryl group, can be a methyl group, or a triazolyl group. Again, I'm just going to show some results. Here, I'm just going to mention we studied the properties of some selected corals, and just mention that these compounds also absorb within this phototherapeutic window, okay? I'm just going to show some selected results. In this case, we focus on lung cancer. And what we observe is that the compounds with this phenyl oxime derivative was significantly more active than the methyl derivative, but the triazoles were the ones that show higher activity. So we then identified derivatives with nanomolar activity and low dark cytotoxicity. So we do think they are also very promising PDT agents. We also expand this chemistry to hydrazone. This is currently also being started with Bruno Costa. He's working in this with these compounds as a PhD project also. So starting with halogenated hydrozones, in this case, you obtain the corresponding Milan, and then the oxidative macrocyclization gave these Courant derivatives. We also obtain a significant range of derivatives, also having different aryl groups at meso positions. The group attached to the hydrazone can be a methyl or phenyl group, but also different groups directly attached to nitrogen. Again, showing some selected examples. In this case, the methyl hydrazones were more active than the phenyl hydrazones, contrary to what observed with the oxime derivatives. But the substituent of the nitrogen is also important. So having the Bock group or Tuzil group will ensure higher activity, IC50 values below 100 nanomolar. With these benzyl derivatives, we've observed lower activity. So just to sum up regarding these Courant derivatives, here are the IC50 values of these oxime and hydrazone Courant derivatives, and compare with this model compound which has only phenyl groups as substitutes. And in fact, this illustrates that the presence of these oxime or hydrazone dysfunctional groups is essential to ensure high activity. This compound, the model compound, show no phototoxicity. In these cell lines, and also in the second cell line study, also a significant difference in activity. The work with on the tetrapyrrolic macrocycles has been carried out in collaboration of the Philomena Bouteille Research Group, with a big involvement of Mafalda Larange. Just mentioned the financial support of these projects by FCT and European funds. Two of these projects as Marta Pinheiro as co-PI. And just to sum up, I know that I have to conclude. So I just mentioned our efforts to develop these new spiral lactams with the host-directed antimicrobial agents. Talk about our efforts also on this novel type of ring fuse screen that can be acted as PDT agent and the corresponding platinum derivatives acting as diagnostic agents. And finally, the family of corals. We also develop a route to this compound, synthetic route, and are promising PDT agents against lung cancer. Just mentioned and thanking my current research group and all the members of the organic chemistry group at the Coimbra Chemistry Center. And just leaving this last slide just with the invitation to come to our next year for the meeting of the International Society of Heterocycle Chemistry, which will be held in Aveiro next year. So we are all invited. And thank you all for listening.
Great. Thank you, Teresa. Super presentation. We're off to a good start, as they say. So the presentation is now open for questions and answers. So there's already a few in the chat. So I'll call them out if that's okay. Two from an anonymous attendee. That's always interesting. How is the antiviral activity measured?
Well, it depends on the methodology. So we study different types of virus. So of course, as I mentioned, my role here is as organic chemistry, and the biological activity is carried out in collaboration with other groups, as I mentioned. Okay, but I can send you information if the anonymous will give the identity. I can give that information later. Okay.
Okay.
It depends on the type of virus, the different methodology.
Following on, I think from the same person, how should one understand the functional group in a heterocyclic system like a beta-lactam? I'm not really sure what that's asking.
Can you repeat that? I didn't understand exactly what you want.
Yeah, I think it's too general a question and it's not very specific. So in the interest of time, maybe I'll pass on to a question from Rafael Gomez.
Okay.
Thank you, Prof. Teresa, for the really nice presentation. I have a question concerning the spiral lactams. Since you are proposing a broad range of activities due to the activation of the immune system, have you performed or considered performing co-culture assays, that is with macrophages or T cells?
Yes, well, we carried out some studies with cells of the immune system, but at this stage, what we can say that they release some soluble particles that show activity against HIV. So we focus on HIV just to try and prove that we elicit, in fact, the immune system. So that's what we're trying to prove now. But what are these particles? I mentioned some proteins related to the immune system, but at this stage, that's what we can say. So there is the release of some particles when we study with the cells from the immune system. So, but we are still working on the mechanism of action, yes.
Okay, perfect. So they're all the questions that came through from the questions and answers. Any questions from panelists? Remember, participants can raise their hand and then I'll allow them to ask the question. But maybe in the interest of time, we might move on to the next speaker if that's all right. So again, thanks, Teresa, and over to you, Arthur.
Thank you.
Well, thank you, Teresa. Can you stop sharing the screen, please?
Yes.
And now we move for Augusto Tomé. Augusto Tomé is from the University of Aveiro, has a long experience in heterocycle chemistry, and today we'll speak about the ketoparols, about the scenes and applications of this. So Tomé, the screen is yours. You can go ahead, please.
Good morning. Are you seeing my
Yes.
presentation?
Yes.
Yes. So, good morning to everyone. I would like to thank Professor Artur Silva for this invitation. I'm going to present some of the work we have been carrying out for the last 4 years. In my group, we typically work on porphyrin chemistry and fullerene chemistry, but more recently, we start to work with smaller heterocycle systems, namely diketopyrrolopyrroles and thiazolothiazoles. And today I'm discuss some of the work we have done with diketopyrrolopyrroles. So I'm going to start to show you what is a diketopyrrolopyrrole, the structure and the properties, how these compounds are made, how they can be functionalized, and this is general ideas. Then I start with the work we have done and some final remarks. So, diketopyrrolopyrroles are compounds with this structure, with two fused lactam units. In fact, when we talk about diketopyrrolopyrroles, we are specifically thinking on this system, 3,4-C pyrrole system. These compounds are very colorful, with a range of colors from yellow, orange to dark violet, depending on the substituents on these R groups or in the N nitrogens. These are why are these compounds important? Because they show strong absorption bands in the visible region, more from 400 to 600 nanometers. They are highly fluorescent with large Stokes shifts. They are highly thermal stable, with melting points above 350 degrees, specifically for the N substituted compounds with the NH groups here. If they have the NH group, they are very insoluble, in almost all solvents, and that's why they are called pigments, because they don't dissolve. But if you introduce substituents here in these positions, they become very soluble in organic solvents, and they call them dyes. So we can work with dyes, or we can work with pigments, and so they are very, very interesting small molecules. They found a range of applications. They start to be used as pigments. Some of them are used in tattoos, so they are not so toxic. Then other applications, photodynamic therapy, both for cancer cells and for antimicrobial photodynamic therapy. This means for the photoinactivation of microorganisms, pathogenic microorganisms. They are being used in electronic devices, namely in dye-sensitized solar cells. A lot of very large range of sensors for anions, cations, biological molecules, and so on. And for bioimaging, because they are very fluorescent. So we can play around with these real groups and the substitutes on the N nitrogen atoms. This compounds were discovered quite recently in 1974. In April 1974, the paper has been submitted in April, in 15 April 74, so it's just before our revolution. It's a curiosity. And the authors wanted to prepare this lactam, and they didn't succeed because they get a highly insoluble, brilliant red crystalline compound with melting point above 350. So that's the compound they get. They were frustrated, very low yields. They published it in a Tetridon letters, and that's it. They move on for their lives. And some guys on the Ciba-Geigy thought that it may be an interesting compound for a pigment, and look at the synthesis and found a new route to the compound, and they patented this called succinic ester route in 1983. So with this can be done in the lab quite easily with a real nitrile succinic ester in the presence of a base, the catalyst, and they get the ketopyrrolopyrrole. One of these pigments became very famous. That's the pigment red 254, which is also called Ferrari red, because it has been used to paint Ferraris. And not so Ferraris, Alfa Romeos, BMWs, Corvettes, Volkswagen, Lexus. So top cars have been painted with this red, solid red pigment. It's a curiosity. Ciba-Geigy was selling this compound for $1,000 per kilogram, and the cost was $20 per kilogram. So they made a fortune only with this compound. So it's very interesting compound. Obviously, we don't have money to buy the Ferrari, the red one, not the yellow one, but we can buy some Ferrari red and we can play with, and we can have fun playing with Ferrari red. So currently 25 grams costs 60 euros at TCI. So with this Ferrari red, we can do a lot of things, namely introduce alkyl groups. It can be lipophilic or hydrophilic groups. So we can choose the groups that we want to insert here. And we also can modify this chlorine atom and use metal catalyzed reactions to introduce a range of other functionalities or functional groups here in this position. So we have two simple sites on the molecules to introduce substituents. Typically, we use alkyl groups, pentyl groups here, or this benzyl group or pentafluorophenyl group, and that will allow us to introduce other groups in this position, as we'll see at the end. Our first work on this Ferrari red pigment was to functionalize with our pentyl groups here, and then introduce boronate ester groups here, pinacolates. This reaction gives a mixture of the mono and the bis-pinacolate, pinacolate. We don't need to separate the two compounds. You just proceed with the reaction with a iodo-aryl or bromo-aryl compound using Suzuka conditions, and we get the mono and the defunctionalized compound. Then we can separate these compounds by column chromatography without any problem. We found that it could be easier to prepare such compounds by a different approach. So we can use directly the chlorines to react with the boronic esters, boronic acids, aryl boronic acids. And again, we get a mixture of the two compounds. And obviously, we can also separate them by column chromatography. The experimental conditions are mild. The yields are not so bad, quite good. And we can, by this procedure, we can insert groups, a range of real groups with other functional units, fermion, ceto groups, amino groups, hydroxy groups, or even heterocyclic systems. So it is a very general route for new decatopyroles. These compounds, as expected, are colorful, very fluorescent, so they have quite large Stokes shifts, absorption bands quite intense in this 500 region, 500 nanometers, and emission at 575 typically nanometer region. So the fluorescent quantum yields are quite high for these compounds, so they are interesting for a range of applications. One of these compounds, the aldehyde, was used as a sensor for total protein determination in urine, and this work was developed here with my colleagues in the Department of Chemistry in analytical group. And they found that the sensor, which is an optical fiber coated with the decatopyrrole, can be used to analyze or measure proteins from 10 milligrams per liter to 100 milligrams per liter. So it's quite accurate and useful. The same compound was reacted with C60 and fullerene and N-methylglycine, and we get these quite large molecules, these decatopyrrole fullerene structures, and these compounds were used for the photoinactivation of Staphylococcus aureus, and the results show that these are very good compounds for the inactivation of these microorganisms. This has been done in collaboration with Edgar Durantini in Argentina. One of the compounds shown previously with a pyridine units was cationized with methyl iodide, and the cationic compounds was used to stabilize G quadruplex of DNA structures, which are structures involved in genomic instability. They are present in cancer tissues and so on. So it's interesting to have good probes for the detection and stabilization of those quadruplex. And the results with this compound were very, very interesting and very promising for the development of G4 labeling probes. The same dicationic compound was also used for the inactivation of Staphylococcus aureus. And it has been shown that after 15-minute irradiation, when potassium iodide is used as a coadjuvant, in 15 minutes, almost all the microorganisms are inactivated. So in comparison with the compounds without potassium iodide or in the absence of light. So these are very good results and show that these compounds are able to generate reactive oxygen species and to destroy the microorganisms in quite a short time. Using this Ferrari red substituted with pentafluorophenol groups, pentafluorobenzyl groups, we proceed to the substitution of these parafluorine atoms with this pyridine, sulfonyl-pyridine compound. We get the neutral mono and di-neutral compounds that could be separated and cationized with methyl iodide, and the cationic compounds were obtained in quite high yields, almost quantitative yields. They are show some water solubility, and they were used for the photoinactivation of Pseudomonas aeruginosa. We can see that the DPPV alone without light doesn't work. With light, it takes about 60 minutes to inactivate the microorganism. But if you add the potassium iodide, the total inactivation is obtained after 30 minutes. So it was faster and it works nicely. The monocationic compound also show some photoinactivation, not complete photoinactivation, but after the addition of potassium iodide, the photoinactivation is faster and total, and after 30 minutes of irradiation, the microorganism is totally destroyed. So this is a nice type of compounds. They can be easily functionalized from cheap commercial compounds. We can introduce a diversity of functional groups and moieties that may increase the activity of the compounds, or we can use these functional modifications to introduce other functional units, namely porphyrin or other, I don't want to talk about this, other structures that can give conjugates interesting for biological application or other applications. They, as explained, they show large Stokes shifts and highly fluorescent, and they can be useful in different fields of applications, biological applications, technical applications. So we are working on this, and this work was done, as I said, in the last 4 years, just for the synthesis and structural characterization by a PhD student, Victor Almodovar. And the biological applications were done in collaboration, mainly with Professor Edgar Durantini from Argentina, my colleague Teresa Gomes, analytical chemistry, Catarina Ramos, Adelaide Almeida from the biology department. So not many people, but very hardworking people. I would like to thank the organizations that gave us the money for the experimental work. And I would like to thank you for your kind attention. So we have time for a few questions, I think.
Yep. Augusto, thank you very much. a very colorful presentation, and I'm sure Cristiano Ronaldo thanks you very much for all of the pigments that is on his many Ferraris, as they say. So there's a question come through from the chat from João Rafael Valle. Are DPP dyes used in organophotocatalysis either in single oxygen generation for oxidation reactions or single electron transfer mechanisms?
It's a good question. I don't know, because I did not look carefully for those applications, but I can't answer to that question, honestly.
Good. So there's no other questions coming through in the Q&A there, and I don't see any participants with their hands up. Yeah, no, we do. We have Paula.
Thank you very much for your presentation, Augusto. We have done some. We are doing some work on DPPs for the DSCSC. So it's a field that we have been working for some years. But when we try to do the alkylation, usually you have one parallel product that is the alkylation of oxygen. Do you still have that type of problem on your alkylation reactions?
Sorry, did you? I was looking at another thing. I wasn't paying attention. Okay. Please.
Okay. Did you have any problems on alkylation when you done the alkylation of nitrogen, having the alkylation also on the oxygen?
Yes, that's correct. All alkyl groups are also formed. Yes, we can use adequate bases and solvents to prevent that problem, which is a problem. We don't need the all alkylated compounds, but you get some all alkylated compounds. Yes, that's true.
Okay, thank you.
Good, thanks, Paula. Oh, there's another question coming through from the Q&A from Jose Vasquez. I have a question. Have you tried to make asymmetrical DPPs? And if yes, how about the results?
Every time we functionalize, we get a non-symmetrical system, because we got mono and the substituted system, so they are not symmetrical. But the question was not directed to this point. But we never tried, but it's published. It's published routes that give selectively non-symmetrical decatopyrrolopyrroles. We never tried. We always worked with Ferrari red. But it's published, yes.
Great, good. So maybe again, perhaps in the interest of time, Augusto, we'll pause there and go to the next speaker. So again, over to you, Arthur, for the introduction.
Well, thank you, Augusto, for your interesting lecture. And now we go for Pedro Góes. In the beginning, I refer that we are the second generation from when we start the organic chems in Portugal. I can say that Pedro is from the third generation, because Pedro is already, it was supervised by Carlos da Foz, that will speak at the end. So it's the third generation of us, and he's involved in chemical biology. It's now in the Faculty of Pharmacy, University of Lisbon, and today we'll be speaking about some bioconjugates using boronic acids, because Pedro, it's like the boron from our periodic table. Pedro,
yeah.
It's the floor is yours.
Okay, thank you very much, Artur. Thank you also, Pat, and all the organizers. Also, very happy and thankful to Artur to include me in the third generation and not the first, which is important. So I guess you're seeing my. Are you seeing my
yes
presentation? Okay,
yeah.
That's good, excellent. Okay, so we'll start. Let me just use my pointer. Okay, as I said, thank you for these. Today I'm going to try to present a bit of the research that we are conducting in Lisbon in our lab, which has 3 or 4 different topics. Today, I'm just going to focus in one of the topics which is related with our goal of designing and discovering new chemical technologies that can help us to prepare more effective and more efficient drugs, namely in the area of oncology. And if you think about oncology, which is obviously a huge problem that I don't need to be here stressing, one of the ideas that we have and the community is that we need to have drugs with higher levels of selectivity. And a way to do that, we need to spare healthy cells from being killed by our drugs, which are cytotoxic, but they are also active against normal cells. So we need to design drugs that are clever and smart enough to select the disease site and the disease cells to deliver our drugs to these sites. And one of the technologies that is more advanced in the pharmaceutical company and also in the clinic is the design and the construction of bioconjugates like antibody drug conjugates. Okay, so these molecules can do this, and the concept is quite simple. Basically, what you have is a protein that is linked to the cytotoxic drug, and the protein is able to recognize antigens on the surface of the cells, and by doing that is able to target selectively the disease sites. Well, in recent years, this has been a very active area of research, not just in the academia, but also in the industry. And here I represent just the recent on the approved drugs, ADCs that were approved. And these are just 12 mostly, and I think, if I'm not updated, only in oncology. These are the ADCs that have been approved. So designing these molecules is quite a challenge in different areas, from biotechnology to medicinal chemistry, but they are also a very interesting challenge for organic chemistry. And here, and I will tell you why, here I represent here Adcetris, and Adcetris is one of these recent antibody drug conjugates approved for Hodgkin lymphomas. And what you have here is your protein. Now, I'm not going to focus on the biotechnology of the ADC. Then you have your linker here, and in red you have your drug. And the reason this is a challenge for organic chemistry is that this linker, which connects both components, the protein and the drug, needs to be smart enough to recognize that is at the disease site and react with the chemical environment that is provided by the disease. And in this case, what you have is enzymatic cleavage of the D peptide that is in this position to release this part of the ADC, and now needs self-emulatively collapses to release the drug. Okay, so basically, you need chemistries to do this. And if you go back to the ADCs that have been approved, what you see is that most of them rely on enzymatic cleavage. All these ones here are all enzymatic cleavage. You have also acidic cleavage, reducible disulfide bonds, but these are not the main area in which that is being explored to do this type of constructs. Mostly enzymatic cleavage is the one that is being explored for these type of releasing mechanisms. And basically what we realize is that we don't have enough technologies, chemistries that allows you to design stimulus responsive constructs like these, which can be applied in drug design, but also in many other type of materials that need to respond to a specific chemical condition for delivering a specific function. So that's our main goal. And as Artur said, we are very much interested in boronic acids, and that stems from our background in organic chemistry and catalysis, in which we were using these molecules as reagents for making new carbon-carbon bonds, namely using Suzuki reactions. But in time, what we discovered was that we, and I have to say, as a community also, we were overlooking the reactivity and the very interesting properties of the boronic acid warhead, which can, as you know, interconvert between this sp2 or configuration and this sp3, one vacant orbital, a charged species like this. Okay, so if you look at the boronic acid, it turns out these are very interesting tools to play in the construction of ADCs and bioconjugates in general. And I will show you why, because you can take advantage of the coordination chemistry of boronic acids, which generate these dative bonds with oxygen and nitrogen oxygen species. You can also have, and this is an area that we have been actively involved, you can use boronic acids to functionalize proteins directly, and you need this type of chemistries if you want to attach your drug to the biomolecule. So you need bioconjugation methods, and boronic acids can give you that. Also, there are a few drugs approved in the clinic which contain the boronic acid function, and I show you just two here, bortezomib and ixazomib, which are first-line cytotoxic agents used in the clinic. So these are all areas that you need to assemble your conjugates. I'm not suggesting, obviously, that you try to prepare a bioconjugate just on boronic acids, but to tell you that these are all very important areas in which boronic acids can deliver in the construction of this type of bioconjugates. Well, another thing that is quite important and very much related with the topic of this presentation is the fact that these heterocycles assembled with boronic acids are pH responsive, meaning that they are dynamic because these BO bonds and boron-nitrogen bonds are dynamic, are reversible covalent bonds, and pH can be used to control this type of coordination. And this is a textbook organic reaction that we typically teach our students, which is the oxidation of the boronic acids in the presence of reactive oxygen species, like peroxides, to generate phenols, and these we know that is typically a very fast transformation. And why this is important for our discussion? Because if you look in cancer, all these mechanisms are very much important. So you have acidic pH, you have higher concentrations of glutathione, you have reactive oxygen species, all playing a decisive role in the disease outbreak and progression. So you can take advantage of boronic acids induced mechanism to design this type of molecules that can respond to the chemical environment of cancer. Well, where did we start? We start by playing with different components, which we know that we can react to generate these type of heterocycles based on boron. And the problem here is that we need, because these are all reversible covalent bonds, we needed to discover complexes that have sufficient stability to operate in physiological conditions, but also that allows us to perform post-functionalization, namely to attach targeting molecules that can recognize the antigens on the surface of proteins to undergo internalization and to release, to disassemble inside cells, now promoted by the mechanism that I just showed you before, and elicit the expected activity. So here just few examples that we have published over the years. This was the first one, which was very much conceptual at the time, which is a boron heterocycle using bortezomib that is glutathione responsive. Later, we extended this platform to include fluorescent also units in this structure. So now we have like fluorescent glutathione responsive conjugates, and these are quite important for us because we are actively developing now this platform, which are pH responsive conjugates. Okay, so today I'm going to tell you about two recent examples in the group, which are the raw sensitive platform. And basically, when we were starting to study these, there were no linkers that could respond to reactive oxygen species. There are several reasons for that. If you want, we can discuss them later. But the fact is that it's quite difficult to generate well-designed and well-defined linkers based on organic functions that can selectively respond to physiological ROS levels in oncology. You can do that with materials, but it's different if you want to design a well-defined linker. And as you know, boronic acids, as I said in the beginning, are quite important in this area because they react, and they is quite fast with hydrogen peroxide and even more peroxynitrile to oxidize to the phenol. And this is not new. Obviously, lots of researchers have used this technology to design fluorescent probes that are sensitive to reactive oxygen species. Christian has done lots of work in this area, but also materials, polymers, and these sort of functions. But if you want to use the boronic acid like this, exposed to the solvent to generate this type of linker is quite difficult because it will tend to oxidize and also interact with endogenous molecules like proteins, carbohydrates, which would lead to off-target toxicity. Well, so we start looking at this, and the idea was, how can we stabilize the boronic acid function so that we can have the response to ROS, but an improved stability in physiological media? And so we went back to the literature, and we were already very much interested in diazoborines chemistry. And basically, these are already old heterocycles in which you have this type of structure. We have an hydrazine that reacts, and basically what you have here is the donation of these electrons from the nitrogen to the boron, which stabilize the vacant orbital of the boron. And although these were quite old heterocycles, recently they became very popular in chemical biology, again, for different reasons that I don't have time now to enter in detail. And this is just a recent example by Dennis Hall's group in which he studied carefully the properties of these heterocycles. Okay, so in our case, as I said, we were interested to understand if we could use diazoborines as linkage. And basically, they formed quite well, simply mixing hydrazines with these carbonyl phenylboronic acids. And this is one of the reasons why they are quite popular now, because this reaction is really, really fast. As you see, almost 100%, around 1 minute or two in the low micromolar range. So what we've done, we study quite a few to look for faster conjugations, but also higher stabilities. And what we found was that these alkylic diazoborines with the hydrogen, not the methyl, can give you very high stabilities in aqueous media over 14 days, no degradation. Also at different pH conditions, you see 7.4 buffer, 9, 4.5, even plasma. We didn't see any degradation of this sort of diazoborines. But then what is quite important is that in the presence of reactive oxygen species, you immediately observe the oxidation to the alcohol. And this was a relatively surprise to us. Why? Because we knew that if we were stabilizing the boronic acid, we could compromise reactivity with the reactive oxygen species. But it turns out that this is not the case. We maintain the ability of the boronic acid to react, and we studied that also carefully in terms of DFT calculation of the mechanism, and is quite similar to the parent phenylboronic acid. Here, just the oxidation rate for you to see. Under 15 minutes, everything was ready. Okay. Then what you see here is the decay of the oxidation with even 10 equivalents. So we reduce substantially the quantity of the reactive oxygen species. And this chart here is quite important. Why? Because in cancer cells, you have reactive oxygen species, but you also have reducing agents like glutathione. And we wanted to see if even in the presence of glutathione, we could still have the oxidation going. And this blue line affects a bit the rate of the reaction, is slightly slower, but still we can have the oxidation going. So this was quite good news for us. So then came the challenge of how we can incorporate this mechanism in the design of a conjugate like this. So the idea, the first design was oxidation of the boronic acid. So we install in this para position a self-emulative linkage. Why? Because once you have the oxidation, it will collapse to generate this methyl quinone and the release of our payload. Okay, so it looks simple, but it was a challenge to design this molecule, namely the simple synthesis of this unit in which we attach SN38, which is a drug used in the clinic, is a topoisomerase inhibitor, and basically we were successfully capable of preparing this molecule. And then what we did, just design the linkage with the hydrazine, and we prepared the diazoborine, which then we study again completely in terms of oxidation. And more importantly, what we observe is that upon oxidation, we also observe the release of the SN38. So that was a very important information for us. We install here an azide to do a post-functionalization to modify the protein, which we use a protein developed in collaboration with our colleagues from the medicinal veterinary faculty. So it's an antibody that recognizes human T cells, lymphoma T cells. And basically in the evolution of this protein, of this antibody, we use the human Jurkat cells as a negative control. So for your reference, canine T cells are recognized by this antibody, Jurkat cells are not. So the ADC was simply constructed as it is here, so I'm not going to delay on the synthesis, but mainly my chemistry to modify the protein. Here, just the mass specs of the intact protein, which are here, and here the mass specs of our construct once isolated. In terms of activity, it's quite interesting. So if we look in this graph here on your left, what you see here is the antibody has no activity. SN38 is quite active in the range of 4 nanomolar, and this is our construct, slightly less potent, around 54 nanomolar. So we lose a bit of potency. But that is also quite normal in this type of evaluation. So the question is, how about selectivity? And in terms of selectivity, now we look into Jurkat cells, which were the negative controls, in which neither of the components are active. The protein is not active. Our conjugate, our antibody is not, antibody drug conjugate is not active. It's here. So everything here, but then SN38 is still quite potent, around 10 nanomolar. So looks like, in fact, the protein is doing what it should be doing. Okay, so you could ask, probably this is a question of different ROS levels between both cell lines, and we checked and confirmed that both cell lines have the same level of reactive oxygen species. We also studied the reaction mechanism, the biological mechanism, and basically the SN38 is an inhibitor of topoisomerase, which typically uncoils DNA for reading. So basically it goes from supercoiled to relaxed DNA when it's not present. And basically if you add SN38, you inhibit this enzyme, and basically you observe the supercoiled DNA, which is exactly what we observed when using our ADC. Okay, so a different design. Is this platform something that we can play with to install different the drug in different positions? So this was our first design, and then we thought about placing the drug now in the nitrogen of the hydrazine. How? Because upon oxidation, we hope to hydrolyze the hydrazine, which then, either by cyclization or other sort of mechanisms, can start releasing our components. So this was recently published in Science Reports, and it took us a bit of time to correctly discover the length we needed to have in this position. We studied different type of linkers until we discovered that the best results could be obtained with one single carbon in this position. So we observed the oxidation, and then we were able to monitor the release of the alcohol using these model compounds. Well, with this, we then move to the synthesis of the ADC. Again, similar principles, maleimide chemistry. Here, we don't need to have an azide to do any sort of additional modification, because simply we will modify the boric acid now with the maleimide. Okay, so you see that we also functionalize a different position of the SN38, and this is exactly the position that was selected recently to design an ADC that is in the clinic against triple-negative breast cancer cells. Troveldi, which functionalizes this alcohol here, and this is even important because it stabilizes also the lactone of the drug. Okay, so in terms of mass spec, basically the same. In terms of cytotoxicity, the selectivity was the same, meaning that is active against B-cell lymphomas, but is not active against Jurkat cells. But to our surprise and delight, also we improved considerably now the potency of our conjugate, so meaning that this design gives us a more potent ADC than the previous one. So we are currently further exploring these ADCs and this technology with Technophage, which is a biotech company here from Lisbon. Just to say again that the mechanism of topoisomerase is exactly the same as shown before. Okay, for the last example that I prepare for you today, I hope I'm still okay with time. So just to remind you that we have developed glutathione sensitive complexes, we have designed pH sensitive complexes, and now we've come up with new raw sensitive platform. Okay, so in this area, light is becoming ever more important to design molecules that can be activated by light in this context. So we wonder, is it possible that we can also use light to induce a response from this sort of complexes? Okay, so recently we have designed and we have discovered these fluorescent Bashi platform, basically based on three simple components: an hydrazone, again, phenylglyoxylic acid, a boronic acid. Simply mixing these three, we can have a fluorescent molecule in almost quantitative yields up to 99%. And we have been exploring this platform, this highly modular platform that you can prepare in gram scale. So I think this is quite useful, and you can diversify in terms of structure in a very simple manner. All of those involved in synthesis of fluorescent molecules for biological application. You know how difficult it is to modify synthetically the cores that we have available. Body pies, cyanines, they are not as straightforward and amenable to modification. Completely different with this new platform. You can also go from red up to green, and here you can have some, these are just some of the properties of these molecules, and here's some images that you can obtain with this type of molecules. We've done this already in vivo, so you can also use Bashi in vivo applications. Okay, so what we know about these molecules is that they have a very high tendency to accumulate in these green dots here, which are lipid droplets. This has to do with their configuration, which are hydrophobic, their 3D shape. They tend to accumulate in these lipid reservoirs inside cancer cells, as you see here. So we thought about this, and we came to the idea that, okay, if BASHs tend to accumulate in lipid droplets, if they can work as a photosynthesizer, photosensitizer, that you have heard already about this technology by Teresa and also Augusto, which are much more knowledgeable about this than I am. I'm just a newcomer in this field, which if the BASHs tend to accumulate in lipid droplets, and if you can use a PDT approach to generate oxygen singlets from these type of structures, because they are accumulated in a lipid reservoir, perhaps you can induce lipid peroxidation, which is an hallmark of ferroptosis. So this was our very recent study that we have just deposited in ChemArchive. And basically what we've done, we prepare a range of BASHI structures with different cores. Here we just wanted to study what type of core with the cyanine type would be more efficient. I'm not going to detain much on the photophysical properties of these molecules, just to tell you that in fact they can produce oxygen singlet in a very effective manner, up to almost 80%, and mostly this type of molecule with 2 double bonds in this position. So we were very much excited with these. All these studies, the photophysical characterization were performed by my colleague and his team in Huelva, Uwe Pichel, great scientist, also a great friend of mine, which is today turning 50. So if anyone knows him, you might want to just send a message to him. He's getting old. Basically 80% quantum yield in terms of oxygen, singlet oxygen production. Okay, so but does that mean that we can use this as a photosynthesizer just because it produces oxygen singlets? So we decided to study these by preparing these 2 types of molecules. One is a simple molecule, the other one we modified the boronic acid component to install a cholesterol. This is important to say that because these molecules are highly modular, you can easily install different parts and different components in a single step. And we introduced the cholesterol Y to try to play with the localization, the intracellular localization of the BASHI. We know that this one accumulates in lipid droplets, and we wanted to study if the lipid droplet oxidation would lead to a more effective ferroptosis. So we prepared this one with cholesterol, which is known also to interact with lipid membranes to divert the accumulation in lipid droplets to try to demonstrate if this accumulation was important in triggering this mode of cell death. Well, so we prepared these molecules, and what we observed was quite amazing for us. These studies were conducted by my also good friend Gilles Gasser in Paris Chemitech in Paris, and he is a well-known specialist in PDT. And what Gilles observed was that these molecules, namely this one here, was really effective against human glioblastoma cancer cells, in which he observed that a potency of around 4 nanomolar. But what was very striking was this very high photo index around over 22,000. Okay, so this is far beyond most of the PI indexes for the known photosensitizers. So this is really quite high, and this gives you a relation between the toxicity of the molecule under light and in dark. So it means that they are really safe. And then when we start playing with this one, with cholesterol, the potency is significantly low, around 98 nanomolar, still a good PI, but not comparable with the one that we had observed before. So what was going on here? It looks like, although we have the same core responsible for the photophysical properties, this part, the cholesterol, is doing something to our molecule. So Because these compounds are fluorescent, we can track their intracellular localization. And here you see this di-A accumulation in lipid drops, as you see, because of the conjugation now is red. Okay, so this is di-A, and now this one is di-B. You observe that is clearly less uptaken by cells, still accumulating a bit in lipid droplets, but the profile is completely different from this one. So Gilles was very intrigued by this, and he conducted very detailed experiments by ICP, because we are using boron. We now can use these. He discovered that di-A accumulates much faster and in much higher yields inside cells than di-B. The di-B here, and even more di-A then accumulates inside the lipid droplets. So he also was able to extract the lipid droplets and perform ICP. So we discovered that yes, in fact, we have the dye inside lipid droplets, but again, di-A is far more effective than di-B. So basically, what we can take out of here is that the fact that now we have the cholesterol, it somehow is inhibiting the internalization inside the cell, also not promoting as efficiently the internalization in lipid droplets, meaning that all that is very important to have a more potent and safe drug. Now, is lipid peroxidation really involved in triggering ferroptosis? It was our hypothesis correct? So to do that, we study what happened in terms of cytotoxicity in the presence of antioxidants, which are commonly used in these studies, ferrostatin, tocopherol, and basically with antioxidants, remember that we are performing the peroxidation of lipids, so having antioxidants can diminish the toxicity of these species, and in fact, in the presence of di-A, which now is the name of tree, in the presence of tocopherol and ferrostatin, we clearly improve the viability of the cells. So meaning that in fact antioxidants are important. Another, and that happens for both dyes, and more importantly, when you use this inhibitor of the cytosolic phospholipase A2 alpha, which is an important enzyme in lipids and in the formation of the lipid droplets. When we inhibit this, basically what happened when we decrease the quantity of lipid droplets, we also increase the viability of the cells. Altogether, I think we have shown quite carefully that BASHs are not only very efficient photosensitizers that can produce high quantum yields of oxygen singlets that accumulate in lipid droplets, and by doing PDT assays with these dyes accumulated in these reservoirs, we can induce a lipid peroxidation, which then lead to this very important mode of cell death, which is ferroptosis. Finally, with colleagues from Canada, we're also able to study and evaluate the toxicity of these dyes in zebrafish embryos, Danurerio, which are very important clinic models to perform a first assay of this type of molecules. And basically, we shown that at concentration, which is the typical concentration to study the toxicity and safety of molecules, which is 10 micromolars, we didn't observe any toxicity of these drugs. And then when we increase and we expose these embryos to light, we start observing very severe abnormalities in the development of these fish. Here in the A, you have a healthy zebrafish, and here you see the abnormalities that are formed once you in this light in these embryos. So basically, this brings me to the end of my talk. And in terms of conclusion, I hope to have induced some curiosity in you to look at boronic acids that we as organic chemists are so much used to use as reagents for synthesis, But to tell you that these molecules they go far beyond simple reagents. The boron functions can be used as a construction tool for chemical biology and for materials to generate constructs that can respond to different stimulus, namely in the area of oncology, as we've shown. Okay, finally, 4 people that were pretty much involved in these studies, Fabio and João Antonio. Fabio was the seminal student and now a scientist in our group that developed the glutathione responsive constructs. João and Joana developed the ROS active platform. Joana already left the group and now is working happily. Fabio and Maria are also responsible for the Bashi platform. Namely, Maria was a PhD student also between my group and Gilles in Paris groups. So she developed the photosensitizers based on the Bashi platform. Also now already concluded the PhD and is working now in the Max Planck in Mainz. Other group members, I have to thank all collaborators that worked with me in many different projects, FCT, MedChem Train, also collaborations with the industry, Ovion and Technophage, and also a big thanks to the Portuguese Chemical Society that has sponsored one of the PhDs working in the group. And obviously, all of you and the organizers for the very kind invitation. Thank you.
Pedro, super presentation. Thank you very much. There are 2 questions after coming through in the chat. The first one from Nuno Tendeas. Apologies for the pronunciation. Thank you, Pedro, for the clear and nice presentation. When delivering the drug from the anti-drug conjugate, there's also the release of a quinone methide. Since such methides are well known to react with biomolecules, did you ever observe some off-target effects, or do you not consider this to be an issue to address?
Okay, thank you. So this is a common but important question, normally asked in this type of questions. So thank you, Nuno, for pointing this one out. Oh, now I see that is Nuno Candeias. Hello, Nuno. Good to see you. So, Nuno, basically, yes, in fact, these methylquinones are obviously toxic, but typically they are used as self-immolative linkage, used already quite extensively in the drugs approved in the clinic. And the reason is that there are 2 reasons not to be concerned about these components. One is that the release is supposed to happen only at the disease site. Okay, so that's why you do all these. The drug that typically is used in these constructs is far more toxic than the quinone. Okay, mostly to start using drugs for ADCs, you are looking at the picomolar, the low picomolar range of potency. So these like incredibly toxic molecules. And another reason is that in aqueous media like these, once you generate the methylquinone, it immediately hydrolyzes to the alcohol. So it won't have enough time to react with the biomolecule. So that's why they are safely used in design of this type of constructs.
Great.
Okay,
the second question from Rafael Gomez. Thanks, Pedro. Great talk on innovative responsive linkers. With all this new technologies and precise control on drug release, any thoughts on internalizing and non-internalizing antibodies?
Yeah.
Are some of the options more promising or both in different ailments?
Yeah. Okay. So this is a debate in the field currently, internalizing versus non-internalizing antibodies. Most of the time, I tend to think that almost all antibodies go through a pass, a moment in which they behave as a non-internalizing component. But basically, the question here is targeting antigens that we know that don't go through the process of endocytosis. And basically, that is important. Why? Because the permeation in the tumor often is not very effective, for different reasons, because of the mass, because of the irrigation of the system. So it's not easy to have these large molecules going through the tumor. So exploring what is known as the bystander effect, which is designing molecules that don't go inside the cell, but rather stick at the surface. Releasing the drug there are interesting because now you are releasing a smaller molecule that is hydrophobic and will permeate in the mass. Okay, so both techniques and both approaches are important. Both approaches are being explored currently. The problem is that now we know more antigens that are internalizing than non-internalizing, but there are already several studies and several ADCs that try to design non-internalizing conjugates. Another aspect is also interesting, which is now as organic chemist, if you think about the intracellular environment of a cancer cell, you have also similar stimuli outside the cancer cell in the tumor site, but they are not as evident and as effective as when you push them inside the cell. You have ROS in the mass, also, of course, but the concentration is lower than inside the cell. You have also an acidic environment, is not as acidic inside the cell. So it's more difficult to do non-internalizing ADCs, in my perspective, obviously. Okay.
Great.
Thank you, Rafael.
Super, Pedro. Okay, so again, we might pause there if that's okay, and I'll pass over to you, Arthur, to introduce Victor. Now that his Zoom has been readily installed earlier today, he's no problems whatsoever sharing his presentation. So over to you, Arthur.
Thank you, Pedro. Excellent talk. And now we go for Victor Freitas. Victor Freitas is a person that works on this type of compounds for a long time. Started first looking at the port wine, where he found a lot of this type of compounds, and then he moves for the synthetic part of them, and now he will present chemical synthesis and application of this type of compounds that were originally found in the port wine. Victor has a lot of experience in the group working on this. He comes from the University of Porto, Faculty of Sciences. Victor, the screen is yours. We are not seeing your presentation, Victor.
I mean, we did about 30 seconds ago.
Yep.
Yeah, it was okay, but now it's not okay. And you need to activate your sound, your micro. We are not listening to you because you need to activate your sound.
So Victor, the mute button is on the bottom left-hand side of the screen. Here, maybe I'll unmute you.
Okay, that's okay.
Okay, now you're
But
there.
we are not. No, you are fighting against Pat. Pat, can you activate the sound of Victor?
Yeah, sorry.
So now it's activated.
Now you're activated.
Yes,
Yeah,
but
perfect.
we are not seeing your presentation, Victor. We are seeing a file with the program.
Okay, let's try. I don't know.
Stop sharing here. Stop sharing and try once again.
Let me try. I have a small problem because I don't see the Zoom
Indications. It's I don't know if it is a button on up depending on your screen. Pat, can you
small square? I don't see something.
But wait. Pat, can you stop the sharing screen of Victor also?
Yeah, hold on. Stop participant sharing.
Something wrong here.
Yeah.
Okay, now it's perfect. Okay.
Oh, now I start again.
Okay.
No, it's the same screen.
Now it's perfect.
Now it's okay. Now it's okay. You can go ahead, please.
I had some problems with the Zoom in the beginning. I need to reinstall the software, but now it's okay. So I start thanks to Artur and Patrick for that invitation, and of course the division of organic chemistry from EuChemS to give this talk. So I lead a group named Food Polyphenol Lab that are involved in the study of chemistry and the physical chemical properties of some classes of polyphenols compounds. We try to explore some potential applications in different areas, such as food science, biological properties, cosmetics, food packaging, and many others. So, and today I'm going to talk about the flavulin compounds, chemical synthesis, and applications, of course, only some examples. Okay. So the main classes of flavulin based dye pigments are the benzopyrrolin compounds, well known as anthocyanins, pyranobenzopyrrolins or pyranoanthocyanins, and pyranostearyl benzopyrrolins. So these two first, anthocyanins and pyranoanthocyanins, are natural compounds that are very studied because of their appealing chromatic characteristics, and also they have some important antioxidant and biological and photochromical properties. The pyranostearyl benzopyrrolin is a synthetic one. It was a very recent pigment synthesized in our lab, and you are trying to find some important applications for this kind of compound. So some applications are in food colorants, in dyes in hairs and textiles. Some applications are as photosynthesizers in solar cells, in photodynamic therapy, pH sensors, and many other applications that could be very, very interesting to explore for this kind of compounds. So anthocyanins and the deoxyanthocyanins, you can see the difference between the anthocyanins and deoxyanthocyanins in that position 3. They are natural water-soluble pigments. They are responsible for the red, blue, and purple color that you can find in plants, flowers, and fruits. And you can see that these compounds differ amongst the hydroxylation and methoxylation pattern of B-ring in that position. And you can have different kinds of anthocyanins: pelargonidine, pyrimidine, cyanidine, malvidine, pturidine, and delphinidine. These compounds are glucosylated in that position 3, and may also be glucosylated in position 5 and 7 with different kinds of sugars. They absorb in the visible vision. You can see here the lambda max of absorption in the visible vision. And the color, and these compounds can have a wide color range from orange, red to more purple or blue colors. So in 2002, we have published a chemical half focused on the synthesis, physical chemical properties, and application that I recommend you about this kind of compound. So the anthocyanins can be extracted directly from fruits and vegetables, and also from some byproducts of food industry. However, sometimes the extractions and the purifications were difficult and very time consuming. So alternatively, there are some synthetic approach to obtain different kinds of anthocyanins. And in that case, to be very useful if you want to obtain a specific family of anthocyanins that we want to study. So the first synthesis of anthocyanins was done by Robinson in 1922, and this acid catalyzed aldol condensation between hydroxybenzylaldehydes and acetophenones. You can see that previous glucosylation of acetophenones with glucose, still glucose derivatives bromine, you can obtain different kinds of anthocyanins if you have different kinds of sugars to acylate in that positions. So apart from the acid catalyzed aldol condensations, involve different kinds of aldehydes with different substitution groups or acetophenons, type B, and type A involving phenols, and alpha beta decatones. You can obtain different flavulent compounds, and you can also obtain these flavulons by Grignard reaction addition using different kinds of substituents, and also by productive transformations of 4-oxoflavonoids by, for instance, by bolivaride in the case of hydroxy chalcones and flavanones, or by reduction by metals from flavones. So you can find very easily different kinds of substrates and reactants to obtain different kinds of structures, and you can have a panoply of compounds to study their properties. So there are many factors that affect the color of the anthocyanins. I start with the anthocyanin compounds, basopyrrolic compounds, and it is well known that the pH of these compounds are sensitive to the pH. You can see in that picture that a very acidic pH, we have a red color corresponding to the flavulin structure of anthocyanins, and with increase of pH, the color fades. And for very high pH, you can see that you have a green-blue color corresponding to anthocyanins. So this can be explained by a reversible process that depends on a pH-dependent equilibrium network. So at very acidic pH, of course, you have the flavulin red cation. With increase of pH, they can undergo a nucleophile attack of water in position 2 to give the hemiketal compound that is colorless. The solution lost the color, which I explain what happened in that picture. And they are in equilibrium with chalcone Cs and chalcone trans. Simultaneously, with increase of pH, you have a proton transfer reaction to give the blue-purple quinoidal base that are in equilibrium with the neonic quinoidal base that have a blue color at a very high pH. So these species are all in equilibrium, but you know that these compounds are very unstable, and in plants and foods, these compounds can be easily degraded at any pH by irreversible process, oxidation process involving oxygen and metal transitions to give some small compounds, small structures like aldehydes and organic acid that are colorless compounds. In that case, that transformation are irreversible. That means you lost the integrity of the structure of this kind of compounds. Here you have the mole fraction distribution of different equilibrium forms in function of pH, till pH 7. At very acidic pH, we have essentially the flavulin cation, and for pH higher than 2.53, you start the flavulin cation start to disappear, the red color disappear, and the colorless compound, the hemiketal compounds, becomes the more important species in solutions. This could be a problem because the flavulin and the quinoidal compounds are the more interesting forms to explore their application in different kinds of industries. So there are some strategies to stabilize the chemical structure and the color and tune the color of these species. One is the chemical transformation by chemical reaction or using biology synthesis. You can obtain different kinds of compounds more stable, that I will speak later, and also by a self-assembled system, for instance, by interaction with dendrimers, micellar systems, and also by inclusion complex with different kind of compounds, like for instance cyclodextrins. You can see that in this kind of complex, the flaviolin structure are protected from nuclear attack of water, so that stabilization improves the red color of the solution. One example is the interaction with dendrimers. Some years ago, we have studied in collaboration with Eduardo Fernandes from the University of Santiago de Compostela, a host-guest interaction involving anthocyanins and a water-soluble polyanionic dendrimer. This dendrimer is a gallic acid triethylene glycol-based dendrimer, as you can see here, that is decorated with another 62 sulfate groups in the end. That was obtained by that reaction by a cyclic addition between the acid and alkene group. You can see here. So the click chemistry allows us to obtain polyanionic sulfate dendrimer. So you have studied interaction of this dendrimer with a solution of anthocyanin. This study was performed at pH 3.5. At 3.5, you have these two species in equilibrium. So you add different concentrations of dendrimer to a solution of amlodipine, and you can observe an increase of the intensity at around 420 nanometers. You have a hyperchromic effect, and you also observe a bathochromic effect. That means a red shift of the maximum of the absorption. Of course, that the increase of the intensity becomes from the protection of the flaviolin structure with the dendrimer that displaced the equilibrium from the hemichetal to the formation of the flaviolin species, and the red shift that you can observe also in that picture with cyanidin, some orange color, and the cyanidin in presence of the dendrimer. You can see that more red purple color. This can be explained by a different complex with transitions that correspond to a maximum of absorption higher than the one for the cyanidin. In this work, we also determined the pKa and the hydration constant for this system, for the anthocyanins alone, and for the anthocyanins in presence of the dendrimer. And you can see that the color of the anthocyanins is much more stabilized, 1 unit of pH in the presence of the dendrimer. The same with the hydration constant, which indicate that effectively the dendrimer stabilized the red color of the flaviolin cation of the anthocyanin. We have also determined the association constant and number of binding sites of the guest by NMR titration. And you have observed that the number of binding sites was estimated to be 295, which indicate that you can have around 2 anthocyanins per terminal unit of sulfate. You can see in that picture, we have the dendrimer here, the sulfate group, and you can see that you can have 2 flaviolin compounds that interact by ionic interaction, and this explains the stabilization of it's an hypothesis, of course, the stabilization of this kind of system. So, change a little bit the subject. I'm going to show you an example of use of catalysis or biocatalysis, transformation of anthocyanins. And in this work, you want to introduce, to form different kinds of acylated anthocyanins with different kinds of fatty acids. So 2 approaches were tested: chemical approach using fatty acid chloride, and a biological enzymatic approach using lipase. The best results was obtained with lipase because lipase was a more specific enzyme, and that promotes the reaction in the position 8 of this sugar, which was more interesting in our work. indeed, you know that synthetic biology is becoming an important approach to make some chemical changes in chemical structures to replace the use of chemicals and reduce the number of steps, avoiding, for instance, group protections, the protections, and many other reactions that could be very useful or painful to be performed. Here you have HPLC chromatogram with different kinds of anthocyanins with different chain length fatty acids, C4, 6, 8, 10, 12, 14, 15. Of course, they were synthesized separately, and you mix together to have this kind of chromatogram. And we have also studied the lipophilicity of this species in a system of octanol water in acidic conditions. And you can see that the solubility in octanol increased with increasing chain length of the acylated group. You can see very easily that the color, the upper phase that corresponds to ethanol, become more dark with the increase of the chain length of fatty acid. We have also studied the stability to lipid peroxidation and oxygen consumption of these compounds. So you can observe that fatty acid conjugation improves the antioxidant activity of this compound till chain length of 8 or 10. So you can see that you have a cutoff effect. And regarding the thermal stability of these compounds, you can see here comparing anthocyanins with the conjugate that at different pH 3, 5, and 7, you can see that the degradation of the conjugate is much lower than that you can observe with the anthocyanin alone. So this data, these results, is very important because they reveal a great potential of application of these compounds in cosmetics industry due to their color, their beautiful, interesting color, and because their hydrophobic nature, they allow them to penetrate in skin issues where they can exert their antioxidant properties. This, of course, is a putative application for this kind of compound. So the next class of compounds are pyranoflavulin compounds. You can see that these compounds have an additional D ring in that position between ring A and C. You have here the proton transfer equilibrium with pKa involved in this system. So our group during the last 2 decades, and as Artur said when he presented me, you have identified many classes of compounds that can be formed in wines resulting from the interaction of anthocyanins with many different metabolites that you can have in wines. Of course, that this happens because anthocyanins are very reactive compounds. It's not so stable, and they react by different mechanisms with many of these kind of metabolites. For instance, you can have for methylvitisins with a methyl group in that position by reaction with the steel. This is a very quick overview. I don't go in details in that reactions. I give you only a small example later, but to give you an idea, with a steel acetic acid, we have the methylvitisine. With this steel compounds, you can have carbonyl compound derivatives. With acetaldehyde ethanol, you can form vitisine B with the H in that position. The reaction with higher compounds like vinyl catechins, you can have a more complex pyranoanthocyanin dimers linked directly to catechins. With cinnamic acids and vinyl phenols, you can have this kind of pyranoanthocyanin phenol compounds. The reaction with catechins directly can have anthocyanin linked to a catechin, or involving, for instance, acetaldehydes or aldehydes, you can have a kind of bridge of between these two catechin and flavian compounds. You can see that the color is different at pH conditions in function of that structure. The reaction of anthocyanins with pyruvic acid here give rise to the formation of vitisine A that was one of the first to be identified in wines. further reaction of vitizine A with other metabolites. For instance, they can react again with cinnamic acid that was already reported here before, give rise to formation of protein B with a double bond in that position. The reaction with acetaldehyde and catechine, you can have protein A that have a very beautiful blue color, and the reaction with, for instance, methylpyranothalcyanine that was obtained previously here, in the beginning, can give to formation of pyranothalcyanine dimers that have a very beautiful sky blue color of pigments. So all these reactions has inspired our group to synthesize many different of pigments, follow a little bit the mechanisms engaged in this process. I can give you an example how anthocyanins react with metabolite in wines, for instance, pyruvic acid. So anthocyanins react with the enolic form of these metabolites. The double bond will attack the carbon 4 in that position, and by a cycle addition process involving the hydroxyl group in position 5, you have the formation of the ring after an oxidation and a loss of water in this process. You can have here 3 kinds of pyranothalcyanins that you can find directly in a wine, and you can, of course, simulate it in your lab. Regarding the equilibrium of the color with pH, we have studied the equilibrium of methylpyranothalcyanins by NMR titration and visible spectroscopy. And you observe that the pKa in this case was 4.6. That is much higher comparing to the pKa of anthocyanins, which indicate that this kind of compounds, the pigment is much more stable to the color is more stable to pH change comparing to the anthocyanins. It was also interesting to observe that the product resulting from the nuclear attack of water that happened with anthocyanins and that form the uncolored or colorless compounds doesn't happen in this kind of compound. So this system is much more simple. We all have a transfer reaction forming different kinds of pigments, both colored pigments. So inspired in this kind of synthesis, Luis Cruz, that is a senior researcher in my lab, and Vania Gomes, it was a PhD in my group. We finished last year. We have synthesized several pigments through a little condensation between hydroxybenzaldehydes and acetons, followed by a cyclic addition involving different kinds of cetone groups. You obtain different kinds of pyranothalcyanins with different substitution groups in that position. After they have synthesized new compounds using alpha, beta, unsaturated ceramic ketones with the formation of those 2 stereobenzyl pyruvyl that after reaction with acetone give rise to the formation of the pyranothalcyanins. You can see that the color is very different between the 2 solutions, and this can be explained by the additional double bond in that position that displays the equilibrium in the visible region to a more red shift changing the color. So one of the application that could be very useful for this kind is this pigment is used as a pH sensor for food pressure monitoring. So we have worked a little bit on this subject, and I will show you what have you done. So, for example, during food spoilage involving microbial growth and biological degradation, we have the formation of many organic compounds and nitrogen compounds like biogenic amines. By that reason, this chemical transformation and the release of amino compounds to the atmosphere, they will change the pH of the atmosphere of packaging. So Our idea, it was to find a pigment that they have a pKa very critical, corresponding to the change of the pH in the packaging during food spoilage. So we have synthesized many different kinds of pigments. Before that was more promising is this one, 1, 2, 3, and 4. You can see that the pKa are different. Some of them are much more closer to pKa critical for the food spoil during the food spoilage. You can see that the color of the two species are very different. That's very important because you need to perceive with your eyes that the color is changing. We have also studied the toxicity of these compounds using cancer cell line, the CACL2, and you observe that the compound 4 is much more less toxic comparing to the other compounds. So based on that, we was prepared the label. So to prepare, the dye was dissolved in a stone solution with cellulosetate and glycerol, placed in a Petri dish as a mold. The solvent was moved by evaporation, and you have the acetate film impregnated with the pigments. So of course that we have performed many physical chemical studies. I don't want to show you these studies, and also some pH responsive properties of this kind of film. And I'm showing you. So we have cut small pieces, small squares of the film. We have put the films in atmosphere in contact with different solutions and different pH. You can see of biogenic amines and ammonium. And we have observed that in the pH between 6 and 7, which correspond to the critical pH change that occurred during food spoilage, you observe this change of the color. So based on this result, you want to do some more real experience. We try it with different types of meat and fish. I will show you our best result with fish. We monitor the change in nitrogen compounds, total volatile basic nitrogens here in this graphic, and the variation of pH during this period inside of packaging of the fish, and also the change in color in the label for 8 days. In this graphic, you can see that the total volatile basic nitrogen increase, which is often used as a biomarker for protein amine degradation, and the pH in second graphic increased simultaneously with the increase of the amino-biogenic, what is supposed to occur. The colors changed observed on the label show a good response for good duration, as you can see here. In that phase, you can consider that you have a spoilage of the food, and you can see that you can perceive by your eyes the change of color of this label. So this very good results, very promising results to use this kind of pigments as a pH sensor for food packaging. My last example, because I don't have too much time, I still have 1 minute, is because of this, the color of pyranotocyanins practically does not change between pH 1 and 5. Flavulent compounds show a great potential for bioinspired dye-sensitized solar cells. So, in this work performed by the group of Fernando Pina, a series of pyranotocyanins were designed first and synthesized and applied for the first time as dye sensitizer. It was studied the influence of different linker dimers, the presence of the catechol groups in that position, and the presence of the carboxyl groups, as you can see here. And the results that are shown in this table, you have observed that the catechol unit B and was shown to be essential for an efficient electron injection of the dye into the titanium dioxide semiconductor. Oppositely, the presence of a carboxyl group, you can see here, the carboxyl group is in C, in this case C and D. You can see that the efficiency is much lower compared to the other case. This is due to withdrawing character of the carboxyl group. You also have here the presence of the N-dimethylamino group, and you can observe that the efficiency is much higher when we have a electrodonating character in this kind of compound. So my last slide, how can we synthesize this kind of amino pyranothianins, differing in the number of double bonds in that position? That reaction can be performed by interaction with 4-methylflavellum with different aldehydes, sample and beta unsaturated, with different substituents. In that case, we have aromatic amino aromatic compounds, and by a cyclo addition reaction, similar that happened in previous synthesis and inspired what happened in wines. You can have different kinds of pigments with different substituents, with different amino groups, and different number of double bonds that will change their capacity to transfer the electrons in the cell dyes. Regarding the mechanism, it's not very clear how they interact. So it supposes the formation of a charge transfer complex between the aromatic aldehydes with electrodonating group and the 4-methylflavellum via ionic or radical mechanism involved in these reactions. So, in conclusion, our goals is to continue to design new anthocyanin derivative molecules, not so anthocyanins, but pyranoflavellum or flavellum derivative molecules, to explore their properties in different kinds of applications, as pH sensors, of course, as colorants, dyes, study some biological properties in cosmetic, essentially for cosmetic, in photodynamic therapy also. So there's a lot of application of this kind of compounds. Develop, understand new mechanism for the color tuning. You can see that you can using different kinds of structures, macromolecular structures, you can change the color and stability of the pigments. It's something very interesting that you want to deep in our studies. Explore, of course, new technological applications, new possibility to use this kind of compounds. And of course, because many of these compounds are extracted from byproducts from the food industry, you can, of course, contribute to more sustainable use of resources of this kind of industries. So the most important are people that are working with me. The part concerning the flavellum compounds is more the work more involved by Luis Cruz. When the PhD is already finished, Joana and Nuno Mateus are involved in this kind of study. Of course, that we have some PhD students that are still working this kind of compounds. Our collaborator, Fernando Pina, Nuno Basílio, Eduardo Fernandes, I have said, and of course, all the sponsors that help us to do this kind of research. And thank you very much. I hope that you enjoy my presentation. And if you have some questions, I am prepared.
Great. Thanks, Victor.
Okay.
Oh, presentation. Your presentation is gone again now. But again, open for questions. There are none coming through currently in the chat. So again, I'd ask participants to raise their hand. Any of the panelists also asking questions? Oh, okay.
Probably no questions. I was very clear.
Yeah, no, it certainly was very clear. So I suppose one question, Victor, for me, if that's all right. So what are the outstanding challenges in anthocyanin chemistry? So what is the unsolved problem, or what does the future lie in this area?
The big change is the stability of the color, is tone of the color of this kind of compounds, because they are very unstable, of course, as I said, and you can have some strategies to increase their stability by chemical transformations, as I show you, and essentially, which I think that could be much more useful, is using self-assembly systems. There's different kinds of like cyclodextrins, I told you, that can form complex that protect the attack of flavulums and toxins from water. Finally, the water is a poison for this kind of pigments. But this is perhaps one of the biggest challenges, is the stability of the color, is the stability of the blue color. It's very important even for many kinds of industry, like food industry and photochemical industry, to stabilize the blue pigments at a very low pH. Because the blue color in general, for anthocyanins, you can have a pH 7, 8, 9, and it's very high, because in general, the biological systems, and systems in general, the pH is more or less neutral, and at neutral pH, you have problems to stabilize the blue pigments. So this, in my opinion, is the biggest challenge people are working with anthocyanins they face in the future.
Oh, fair enough. Thank you for that. Again, any other further questions? I don't see them coming in the chat from other panelists. So we might come right back on time if that's all right. So Victor, thanks very much for your presentation.
Thank you.
Over to you, Arthur.
That's fine.
Thank you very much, Victor, for your presentation, excellent presentation. Now we move for the south of Portugal once again to Paulo Branco from the University of Lisbon. And well, this is a combination that I referred at the beginning, the natural products and the synthetic organic chemistry. So it's an alkaloid that was isolated by this group, and now they are trying to synthesize them by asymmetric and non-asymmetric versions. So Paula, the screen is yours. You need to activate the sound.
Okay.
Yeah.
The presentation in Zoom.
Not yet. Yeah.
Okay, now it's done.
Is that you can go ahead, please.
Well, good morning to you all, and thank you, the organization and Artur, for the invitation to be present at this seminar, and to be able to present one of the projects that we have been involved these last 5 years. And it concerns the total synthesis of a molecule that we have isolated on the group a few years ago. It has an aminal core. And as we are developing that synthesis, we faced a lot of challenges. And as we were overcoming those obstacles, I can say, we got some insights on the stability of aminal-based molecules. Here on this slide, I have some selected examples of natural products containing aminals. And here, as you can see, most of the 2 nitrogens from the aminal are belonging to 2 fused cycles, which confers stability for this type of molecule. There are, besides the conventional one, to the conventional method to prepare aminals that relies on the reaction of aldehydes with the amines, there are a lot of other methods, and here I have categorized them as a metal catalysis, as this one that have CH activation by ruthenium of this hydroxymic acid. metal-free alternatives as this one that has chiral phosphoric acid to do the catalysis between these two reagents, some decarboxylative coupling special for amino acids, and there are also some photoredoxes approach here with an example of isoquinoline photoredox catalyst. And also there are some rearrangements. Usually they are applied for the synthesis of peptidomimetics, and they rely on the Curtius-Hoffmann on Lawson rearrangement. Or cernimidine was isolated by the group a few years ago from a plant from the south of Brazil, Solano cernuvel, and it's widely used in traditional medicine. It has a very interesting biological activity, and here I highlight the anti-inflammatory activity by inhibition of the production of interleukin 8. It has as a characteristic, structure characteristics, what we call an hybrid aminal core, because it has one of the nitrogens inside of a cycle and the other one it's exocyclic. It has high potential for chemical modification, and it has low molecular weight. It's a very simple molecule, and as you can see, it has only one stereogenic center. But although very simple, it was really a challenge to achieve this molecule, and I'm going to present the first racemic approach and then the enantioselective approach that we were doing these last few years. So a proposed cernimidines biosynthesis relies on the decarboxylation of L-arginine following this intermolecular cyclization to the cycloaugmentin, and then cycle condensation with an unity of isoferrulic acid. And so, having nature as an inspiration, we decide to approach our synthesis by doing the decarboxylation of L-arginine. And when we tried this with several reagents, we didn't have any success on the reaction. So we decide to do the derivatization of the L-arginine previous to the decarboxylation step. And for that, we have prepared several derivatives, usually conventional methods. And here I highlight this dipeptide, and this is the set of compounds that we have prepared, the L-arginine derivatives. And so picking up these compounds, we decide to apply first the Boto conditions that use diazoxyiodobenzene and iodine to do the decarboxylation. And after trying several conditions, we saw that the best solvents was indeed acetic acid, probably due to the solubility of our starting material. We were able to obtain our compound in a good yield in 3 days at reflux, but when we try other substrates, the yields were not very encouraged. So we decided to apply another method that is also described on the literature for the decarboxylation of amino acids, that is the silver nitrate and ammonium persulfate. And after trying several reactions, we observed not only the formation of our 2-amino pyrrolidine carboxamidine core, the one that we wanted, but also these lateral products, the imide, the aldehyde, and the benzamide. So we tried several conditions, and we have observed that adding an additive copper sulfate previous to the decarboxylation, we were able to obtain this compound in very high yield in half an hour only. So the mechanism here relies on the decarboxylation and the formation of this imino intermediate, intermolecular cyclization to our core compound, or by the addition of water to the hemiaminal, next oxidation to the imide, or cleavage of the bond here to achieve the aldehyde and the benzaldehyde. So we think that here the copper has a role that is to do a complex previous to the decarboxylation between these two nitrogens, and after the decarboxylation, the cyclization goes faster. And so all of our reaction need to have the copper sulfate previous to the decarboxylation step. We decided to apply this methodology for the other substrates, and we were only lucky to obtain the compound with these 3. When we had on the R group extended peak conjugated systems or benzylic positions, we were not able to find our compound and other analogs of this type. And so here we think that the problem is that it's a radical that is formed and then it's transferred to a more stable position, that it's the benzylic position. So if we were not able to synthesize the cernimidine, we decided to try another approach. And for that, we use proline. and we have derivatized proline with the carboxamidine group, and then after doing this carboxylation, trap the intermediate with the amide. We have observed on this reaction the formation also of this lateral product, the piholidone, and also the amiaminal. But we have circumvented this by the addition also of copper, that in this compound is only formed in the absence of copper. But with copper, we were lucky on having our compounds being formed. And also the best catalyst that we observed, it was amber leads that allow us this equilibrium to go to this iminion. So we try also the trap of this iminion by other nucleophiles without success. Here we think that the use of other nucleophiles is limited to the stability of the end product and the acidic conditions that are only compatible with non-basic nucleophiles. So applying these conditions to several amides, we were able to obtain those derivatives that we could not found by the allergen method, and also we got cermidine in a 22% yield in a racemic form. So having this done, the racemic approach, we decided to try an anti-selective approach for this alkaloid. And for that, we have done a retrosynthetic analysis. And here we can think in a neck disconnection or an amide coupling. In both cases, we have these two compounds, A and B, that came from the isocyanate hydrolysis. A from the hydrolysis of the isocyanate, and B from the Grignard addiction of the isocyanate. The key step on this synthesis is the Curtis rearrangement, and for that we need an acylazide. And at the end, we have L-proline. So picking up this heterosynthetic analysis, we start the synthesis by introducing the carboxamidine group on the piholidine with success, using this pyrazole carboxamidine reagent. And then we try to do the acylazide using as conditions the mixed anhydride using N-methylmorpholine and isobutyl chloroformate. But as soon as the mixed anhydride is formed, we have an intermolecular cyclization, and this intertwin is formed instead of our acyl compound. So we have revisited our retrosynthesis and decided to leave for the last step of the synthesis the guanylation. The rest of the retrosynthesis is the same, having as key step the Curtis rearrangement. And so picking up proline, we use N-methylmorpholine and isobutyl chloroformate to do the acylazide, do the rearrangement in THF at reflux usually in 2 hours, and this is followed easily by infrared by the disappearing of the acylazide bond and the appearing of the isocyanate. And then we have screened several conditions to do the hydrolysis. And we have observed that using acidic or basic condition on the hydrolysis and then trying to trap the intermediate with benzoyl chloride or benzoic acid and the coupling reagent, we were only able to find the formation of 3. But when we have done the hydrolysis in neutral conditions only in water and then do the reaction with benzoyl chloride, we obtain this compound but in very low yield. So this emphasizes the stability and short lifetime of POR, which really reacts with the isocyanate as it's formed, and so the urea is indeed the major compound. So we went to the other approach, that is the trap of the isocyanate by organometallic reactions. Our first assays were done with phenyl magnesium bromide, and as we have observed that as we lower the temperature, we increase the yield of the reaction and also the enantiomeric excess. But if we leave the reaction more time at -41, 2 hours or 3 hours, we decrease the enantiomeric excess. The same type of trend we observed with vinyl magnesium bromide, that when we lower the temperature, we increase the enantiomeric excess. When you use these conditions -41, 1 hour, with the methyl magnesium bromide or butyl lithium, we have observed that the reaction was not very famous, low yield, probably due to the instability of the end product, and the enantiomeric excess was 0. But when we use phenylithium, we got our compound in 42% enantiomeric excess. So we tried to rationalize these enantioselective results, and for that we thought that maybe we could have the release of the isocyanate forming this intimate ion pair, and then recombination, and then the stereogenic is formed, but in the racemic form. But if this was the case, we would see the same type of results for all the substract, which was not the case. So we propose two fast ways for the immuno-induced racemization. We have here the pair of electrons from the exocyclic nitrogen doing the ring opening of the pyridine ring to do this zwitterionic species, or having the endocyclic nitrogen doing the release of this amide to do this intimate ion pair. We had these results to summarize our previous table. So if this was the case, fastway B, we would see a different result because more acidic amides will be more prone to elimination. So assuming that we have fastway A here going on, we can think that the racemization can be the result of the quenching of the reaction by the proton source. And for that, we, or in the other case, by the disintermediate is formed by the addition of the metal to the to the organometallic to the isocyanate. Going to the first hypothesis, we sought then to do the reaction in toluene, that it's a polar solvent, and we observed here more or less the same type of enantiomeric excess. Then we tried to see if we could have some type of kinetic isotopic effect, and for that we have done the quenching of the reaction with deuterium methanol, but the enantiomeric excess was more or less the same. We lowered the temperature to see if we could diminish the rate of reaction, but in THF at -78, we got the same type of enantiomeric excess. So we went to the second hypothesis. When we do the addition of the organometallic, we may have an intermediate by the metal coordinated with the oxygen that is more favorable, more electronegative, or we could have the metal coordinating with the nitrogen that could help the ring opening of the pyridine ring. Indeed, when we have done the reaction in phenyl lithium, we see that we have some type of hole of the metal on this type of results. We have also tried other scaffolds, and for that we have prepared the piperidine and the indolene derivatives, doing the picking up the acids, the silicides, the isocyanates, and the trap of the isocyanate with the phenyl magnesium bromide. And we have observed in this case that we have a racemic compound here, and we have 100% enantiomeric excess with this indolene derivative. So we decided to try computational studies and to have some light on these results. And we have calculated the energy of intermediates for the addition of phenyl magnesium bromide for the isocyanate. And we saw that the coordination of magnesium with the nitrogen exocyclic is a more stable intermediate than when we oxygen of the amide coordinating with the magnesium. But then we found two other intermediates, one that has magnesium coordinating simultaneous with both oxygens from the amide and from the carbamate, and the one that is forming an 8-member cycle, and the one that has the magnesium coordinating with the nitrogen and the oxygen of the carbamate, forming a 6-member ring, and this is indeed the more stable intermediate that was found. From this intermediate, we go to a transition state where we have magnesium tri-coordinated with both nitrogen and oxygen, and at this stage, it happens the ring opening to this imine intermediate, and this imine intermediate then through a transition state that corresponds to the rotation around the C1-C2 bond leads to the other isomer, and then again refreshing the pyridine ring by intermolecular cyclization. We have the other enantiomer. So we look for computational studies for these results, and we have as an explanation for the 100% enantiomeric excess of this reaction that the rate limiting step here, it's not the ring opening of the pyridine, but the rate limiting step is the rotation around the C1 and C2 bond, and that's why we have here 100% enantiomeric excess. Having this result, we decided to do some experimental results that can correlate our computational studies, and for that we have prepared this intermediate, this compound that has an orthometoxy group here that we thought could coordinate even more with magnesium, stabilizing the intermediate 1 and allowing the ring opening more easily. And if this was the case, we would see a decrease on the enantiomeric excess. And indeed, we have prepared this compound as usually, and we have observed the formation of our 2-amino pyrrolidine compound in 5% enantiomeric excess. Next, we thought we have to remove the carbamate group and have here a group that cannot coordinate with magnesium. And for that, we have prepared these compounds and the methyl, the benzyl, and the phenyl derivatives. And we have done the silicides of these compounds and have done the rearrangement. And we have observed by infrared that until that step, it went very well, because we see by infrared the isocyanate band, but as soon as we add the organometallic reagents, we had the decomposition of the compounds, so we were not able to prove this hypothesis here. Having established that we have a racemization going on the addiction of the organometallic to the isocyanate, we had to proceed to our total synthesis. The next step was the EC reaction. So we tried with palladium acetate and triphenylphosphine, we tried several reaction conditions. Acetonitrile, the yield was only 25%. We put acetonitrile in the seal tube and we increased the yield to 50%. Then dioxane also to 60%, but in dioxane in the seal tube, we obtain our compound in 84% yield. And so we decided to use these conditions to prepare our substracts with the Bock group on the pyrrolidine. And here we have observed that with the compounds, we usually have a decrease on the enantiomeric excess on this EC reaction. Here I have the example of the parafluor that we got in 20% enantiomeric excess, and we have observed by X-ray the presence of both enantiomers. So most of the compounds have really a decrease on the enantiomeric excess. So we look for the stability of this compound in the EC reaction conditions. And this compound alone in dioxane in a seal tube, it racemizes in 4 hours. When we have palladium on the medium without our alloarine, we observe a small decrease on the enantiomeric excess. So here we have another step that is not helping the enantioselective cities. I have here the precursors for cenomidine. The yield is higher, the enantiomeric excess also, but because we start with the compound with a higher enantiomeric excess to prepare this substrate. So having this step, we had to proceed for the next one. That was also another challenge, that was the Bock removal. And for that, we apply conventional methods, TFA, hydrochloric acid, to do the Bock removal. Here we knew that at the end we would have a free aminal that could be very unstable. And indeed, in these conditions, we observe the formation of the salt of our aminal in the presence of always benzamide. And we decide to try other condition with iodine, that is reported to do the Bock removal. We have only unreacted starting material. With this Lewis acid, we have only benzamide. But then we decide to try another method on the literature for aminals, that is trimethyl aluminium. And trimethyl aluminium at -78 in 4 hours, by TLC on the reaction, we observed only the formation of our compound, the aminal, but we had to do the workup of the reaction, and then we see a small amount of benzamide at the end. So having the free aminals in the form of salt or not, we decide to proceed for the last step of the synthesis. That was the introduction of the guanylation, the carboxyamidine group, using these guanylating reagents. And we choose these 3, although there are a lot on literature, the cyanamide, this pyrazole carboxyamidine reagents. This one has the Bock protecting groups on the nitrogen. And we start first of all with this salt of our aminal, and we need here a base to remove the salt, and here we observe completed decomposition of the reaction. But when we use the free aminal that we prepare and from the Bock the protection with the methyl aluminum and proceed immediately after the workup to the guanylation reaction with these three guanylating reagents, we observe that this one indeed has a low reactivity and needs harsh conditions. And these two gave although three gives a little bit of higher yield, we decide to apply for all substract 2 because here in this case we don't have a later stage of the protection, Bock the protection. And so we were able to synthesize all of our this set of compounds. Cernomidine here in its racemic in its enantiomeric form in 70% enantiomeric excess. Unfortunately for the other substracts, we were not able to find an HPLC program that could allow us to see the enantioselective results of this reaction. And so having this set of compounds and knowing that cernomidine has interesting activity, especially in the capacity of inhibiting the production of interleukin 8 that is associated with anti-inflammatory processes and cancer progression, we decide to study our compounds, the guanylated compounds. So here for the right side of this dashed line, I have the guanylated compounds, and here for the left side, I have the precursors, some Bock precursors of these guanylated compounds. And as you can see here, cernomidine, comparing to its Bock precursor, it's more active. Also this compound that has the butyl chain, it's more active, the guanylated compound, this one, than its Bock precursor. The less active are the ones that have orthonitro and meternitro subsequence on the aromatic substitution here, and the simple dynamic derivative. The highest activity were found for these two. They are very promising compounds. So here I present synthesis of these 2-amino-pyridine carboxamidine core. We present a racemic and an enantioselective approach for this nucleus. We have some insights on the coordination induced aminal racemization and aminal stability of this type of compound. And we found that these compounds are indeed novel candidates for anti-inflammatory therapeutics. As future perspectives, we should examine the impact of the addition of organometallic less basic reagents such as those based on the copper and the zinc on the racemization. We should screen other catalytic systems for that reaction so we could do the reaction at a lower temperature, avoiding racemization in this step also. And so we should do computational studies on the energetic barriers of ring opening on substrates with non-coordinating groups at the nitrogen, those that we were not able to obtain by experimental results. This work was recently presented. Rafael has recently presented its PhD thesis in last July on the enantioselective synthesis and also on the L-proline decarboxylative work. He had the help of Flavia on the studies on the Bock the protection and also guanylation reaction. Luis and Monica were involved and on the L-arginine decarboxylative approach. I have to acknowledge the help of Miguelina Georgieva for the computational studies, Dr. Clara Gomes for the X-ray data collection, and Dr. Catarina Roma Rodrigues from our neighbors, the Life Science Department here at FCT for the biological assay and also the funding. And thank you all for your attention.
Thank you, Paula. Superb presentation. Again, open for questions, and we already have one coming through from the questions and answers. Again, from Rafael Gomez, very busy on the questions this morning, Rafael, very good. Great talk on the formation of these interesting acyl aminals. Have you performed the reaction in the presence of cyanoborohydride in order to trap the potential imine intermediate, thus further corroborating the proposed pathways? And the second bit, also, if you stir one of the enantiomerically pure enriched aminals with the Lewis acid, do you get racemization? So there's two questions there.
This is a very good question. Indeed, with sodium borohydride, I don't recall, but I think we have not done it. Although we have another approach that involved the pyrrolidone, And with the pyrrolidone, we have tried the reduction and do the imino, but not the question of Rafael was to trap the imino, and we have not done. This is a very interesting compound that it has a big question, that it's instability. So I spoke here about the instability of the Bock precursors and also the aminol. But we have also observed that at the end product, when we have the guanylation there, the compound in methanol, after 1 week, it racemize. Bock precursor does not racemize. This is something that we observed, and also from the compound when was isolated for the first time by our colleague here on the FCT, she observed that the compound had an optical rotation, but when she left the crystals to settle down in methanol solution, and when she measured the optical rotation of the compound, and when she has done the X-ray, she saw both enantiomers on the compounds. And so this is something that we have also observed. And now we need to do some computational studies also on the end product, the guanylation at the end, the product at the final, to see the stability. Because here we have also a problem that it's probably the proton transfer and the ring opening that can happen easily, because we observed that in methanol. So to use this compound further, these biological assays were done with we think racemic compounds, those that we were not able to check the enantiomeric excess. We know that in time they can racemize. So this is something that we have to look for, another core that has the aminal guanidine that can be more stable, like for the example, the one with indolene that I showed, that can avoid the racemization. I hope I have answered the question.
Yeah, no, absolutely. And certainly there's no doubt those aminals are tricky species. They're prone to ring opening, and as you say, racemization. It's hard to control what they do. So, you know, it's a very tricky area.
I have a question for Paula.
Sure.
Yeah, nice talk. The problem is the Heck reaction and the racemization, but could you consider maybe an aldol reaction that would give an alpha beta unsaturated?
Yeah, we tried this. We
Okay.
tried it. You tried it, but we didn't follow.
Okay.
With a lot of other thousands of reactions that did not follow. And thank you, Anthony.
Good, thanks, Anthony. Any other further questions? I think I've dealt with the one on the Q&A. So we might keep back on schedule, which is great to say. Let me see. Anthony, you're next. So just from background, Anthony of course is Irish. Anthony Burke is not a Portuguese name, that's for sure. And when I was finishing my PhD in the lab in Dublin, in University College Dublin, Anthony was also in the same lab, so we know each other from then. I was a postdoc in Oxford, and Anthony was a postdoc in Oxford. He with Steve Davis, me with John Brown. So we kind of followed his career ever since. So I'm looking forward to hear your recent results. So over to you, Arthur. Sorry, now I've said a little bit of an introduction, but that might not be known by other people as well. So I said I'd say that as well.
Thank you, Pat. Thank you, Paul, also for your very nice presentation and lecture. Anthony, I know Anthony for a long time, since he came to Portugal. Now he will jump from, he was until recently in University of Évora, and now we moved for the University of Coimbra in Faculty of Science, in Faculty of Pharmacy. And Anthony is well known also by the catalysis, since he organized in the last 10 years excellent congresses in the University of Évora. And today we'll speak about modulation diversity in synthesis using catalytic multi-component reactions. Anthony, I think you will present very nice results on these multi-component reactions. Can you go ahead, please?
Okay, thank you very much, Pat and Artur, for the kind introductions. I'd like to say congratulations to Pat for organizing this great event, and also to Artur for inviting me to give a talk here today. Okay, so let me just share the screen. Okay, is it?
Not yet. We are seeing the Now it's go. You need
Yeah.
to put in presentation mode.
Okay. Okay, presentation mode, and let's go back.
Yes, 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 yes. Go ahead.
Okay, so the title has already been given, so I just jump straight ahead with the introduction. I just talk about multi-component reactions and a bit about chemical space, and I'd also like to talk about particularly the catalytic Ugi reaction and some catalytic petasis reactions that we've been doing over the last 3 or 4 years. Okay, so jumping first to the aspect of chemical space, if you look at the chemical universe, the scale of it is immense. The possibility is up to 10 to the power 63 small molecules to be produced. And that compares vastly to the number of stars in the universe, which is 10 to the power of 24. And in the Merck database, they have predicted about 10 to the 20, or they have 10 to the 20 virtual compounds. But in reality, there's only 10,000 small molecules available. And a very important comment was made by George Hammond in 1968, and I'll rephrase what he said. The most fundamental and lasting objective of synthesis is not the production of new compounds or structures, but the production of properties. And in fact, Barry Sharpless has been hammering this in for the last 10 or 20 years with the development of click chemistry, which is based around this. But today I won't talk about click chemistry. We also are very interested in click chemistry. I'll talk more about multi-component reactions. Now, if you first look at a type of compound called isotene or oxindole, many important biologically active compounds contain this unit. And you just have to look at this one here, which is a marketed drug, and this one here, sunitinib, is used for cancer. They all contain this oxindole or isotene unit. You've also this ACHE inhibitor, and also you have below some preclinical molecules. Okay, and looking at multi-component reactions, okay. When you look at multi-component reactions, I'm just going to expand the screen if I can, and it's not allowing me here. Okay. Okay. Try and amplifying this is a bit complicated today. Generally during lectures, no problem. But okay, so if you look at the history of multi-component reaction, the Strecker reaction is the oldest. It dates back to 1850. Very important reaction. Then you have the Hansch reaction, Biginelli reaction, all in the previous century, in the 19th century. Then you have Mannich reaction and Passerini reaction. And Passerini reaction, developed in 1921 by Passerini, is the precursor to the Ugi reaction, which I'll talk to you about today. The Bucherer-Bergs reaction is something that I will mention at the end of the talk. And then one of the most recent multi-component reactions, the petasis reaction. If you look at the growth of these reactions during the last century, you notice that has been growing exponentially. And when you look at this pie chart over here, you'll notice that the reaction that's most common is probably the Mannich reaction, followed probably by the Ugi reaction. And Biginelli is also quite important. Okay, and what is the impact of these reactions, multi-component reactions, on pharmaceutical development or API development? Well, the Ugi has the main input, basically. So most of these well-known drugs, clobidogrel and lidocaine, have been developed, and many others have been developed using the Ugi reaction. You also have the petasis reaction also being used to develop these types of drugs. And then you have some other reaction, multi-component reaction, like the Hansch and the this Asinger reaction also have been developed some important API molecules. Okay, considering the Ugi reaction, as I mentioned, is probably not that old. It dates back to 1959. As I said, it is a continuation of the Passerini reaction. In the Passerini reaction, you take an isocyanate and it reacts with a ketone and in the presence of a carboxylic acid. You can use a catalyst, but sometimes the carboxylic acid can do that job, and it forms an alpha acyl oxyamide. In the case of the UGI reaction, you have a fourth component, and the fourth component is an amine. And that produces some important molecules, these diamide compounds, as you can see at the bottom of the screen. Okay. With respect to the mechanism, I won't go into much detail on the mechanism. Basically, what happens is you have a condensation reaction between the amine and the ketone. Then you have this ternary complex formed. It forms this type of intermediate here, this acyl-oxy intermediate. This intermediate then cyclizes to form this cyclized intermediate. With protonation here on the imine nitrogen, you get this mum rearrangement, and the mum rearrangement gives the diamide product. Asymmetric catalysis is very important because it produces the drug in a single enantiomeric form, which is crucial for the pharmaceutical industry. So studies into the asymmetric aspect have been ongoing, and some great successes have been achieved in the last 8 years. So a Chinese group led by Zhang showed that you can use this chiral type of Ben-List type catalyst, phosphoric acid, and he achieved remarkable ease up to 90% it's an intramolecular UGI reaction. And one report more recent was this again by Zhang, reported in Science in 2018. And he showed that if you use this spirocyclic chiral phosphoric acid, very interesting, very heavy chiral catalyst, organocatalyst, you can achieve ease of up to 94% and very good yields. The second reaction here on the right is a bit problematic in the case that you need to use dichloromethane, which is not a very good solvent from an industrial point of view, and low temperatures, minus 20 degrees. Okay, so I had the opportunity to write a grant proposal for a PhD student. And in the grant proposal, I had the idea of amalgamating the UGI reaction with isotenes, considering the usefulness of the isotene unit in medicinal chemistry. So I had these ideas where I'd like to incorporate maybe a ketone here as an appendage on the isotene, and then try and do some. Obviously, you have to protect the 3-carbonyl group, which I'll explain in a few moments' time. That needs to be protected before you do any UGI reactions, and then carry out the UGI reaction, obviously a catalytic asymmetric one, followed by deprotonation, and that should give this type of very interesting, highly diverse group of compounds. The other strategy was to include carboxylic acids and also carry out UGI reactions, followed by deprotection. So that was the idea. Then I put the student with Pedro Brandão, who is now a professor in one of the universities of Lisbon, Egas Muniz, an auxiliary, an assistant professor. And so we started trying to put these ideas together. So our strategy was to go and try and put a carboxylic acid unit here on this side of the molecule. Unfortunately, Pedro encountered some difficulties in trying to access this type of compound. We tried another route, as the Americans say, and the idea with them was to hydrolyze this to the carboxylic acid. Again, that wasn't very favorable. So Pedro went to the literature, and he came across 2 important papers. One was by this guy, or this group, Italian group called Rinaldi in 2018. And they were actually doing the UGI reaction where we're using the 3-carbonyl as one of the important units. It was the ketone unit, basically, in the UGI reaction. They got some very nice results because they were forming these types of lactam, diamide-type compounds, using, in this case, trifluoroethanol, in this case. And then there was another 3-component reaction, which was also reported a bit earlier by Lesma, and they also showed the utility of a kind of an imine isotene derivative. Okay, so Pedro then decided that he would like to expand on the work of Rinaldi. But he would like to use different types of catalysts instead of using TFE as his catalytic source. So what he did was he basically started doing baseline reaction with these components here, the isocyanate, an acid, carboxylic acid with a chlorine in the alpha position, aliphatic amines, and this methyl isotene. When he did a reaction without a catalyst, and the ratio was 1, 1, 1, 1 for the 4 components, the yield was only 15%. But he did a bit of a background check on catalysts for the Yogi reaction, and indium chloride came up as one of the useful catalysts. Now, in the next slide, I explain why indium chloride is so useful, but he got some nice results here. He got 65% yield. Okay, so indium chloride is quite useful. It's not a very important catalyst, but it has so many advantages. It's safe, cheap, easy to use, and versatile. Okay, so he did what would you call, a number of reactions and created an extensive library of very interesting diamide compounds based on the Yogi reaction. And some of them, a number of these compounds were then tested for anti-cancer, anti-tumor activity. And this one, for instance, in the middle, this compound here showed a GI50 in a HeLa cell line of 0.27 micromolar. And this one here showed a activity of 230 nanomolar. Okay, and this was tested by a colleague in the University of La Laguna in Tenerife. So lactams are also quite important. I just referred to lactams in respect to the Ryanoldi work. Lactams are present in many biologically active compounds and drugs, like this one, Rolipram and Ropinrolol. They're all compounds based on lactams. So Pedro was interested in focusing on some different lactams that Ryanoldi hadn't developed. So he wanted to focus on oxindole, gamma-lactam hybrids. So the mechanism is pretty straightforward. In this case, you form the imine here on the isotyne. Then you have this cyclization to form, in this case, this type of 7-membered spirocyclic intermediate, and it undergoes the Mumm rearrangement to form the lactam in this case. So he did some screening work, or not screening work, he did scope development, and he produced a nice library of compounds. Now, I won't go into the biologic activity aspects of these molecules, leave that for another seminar, but he got some interesting results, good yields, and very nice diversity. So the point I'd like to hammer home here is the diversity he was producing with these compounds. Okay, so we did some more Yogi reactions, and Carolina Marx, who's an independent researcher in Évora, who worked with me when I was in Évora, also developed some Yogi reactions. Now, she looked at another aspect. Again, chemical space. We were looking at mapping out new chemical space here with isoteins. And she took this amino group, and she wanted to develop the Yogi reaction here around this amino group. Obviously, you need to protect, again, the tree position. And she did a preliminary screen using these conditions here with this isocyanate, with this carboxylic acid, acetic acid, and this isocyanate. And she produced this compound. With no catalyst, the yield was 23%. Now, indium chloride didn't work so well for Carolina, so it worked out at 12%. She did a catalyst screening. She likes really doing catalytic screening work. And she found that the best was zinc fluoride, which increased the yield up to 90%. That was a very interesting result. We're not exactly sure why the zinc fluoride worked the best, but we'd imagine it's the fluorine and the zinc that we have a large hydrogen bonding network involved in the mechanism, and both zinc and fluorine can have a tremendous effect there. Now, one nice reaction that was developed by Caroline is this reaction here. It's she could convert this bromoarine to the aniline using copper iodide, L-proline, and sodium azide in heating. So that's a very useful reaction because some of these building blocks are quite expensive from the usual chemical suppliers. So she looked at the diversity aspect of the reaction. So she looked at the isotyne end side chain diversity, and she could get these types of target molecules in good yields up to 70%. When she looked at the isotyne diversity, she could also make these types of compounds, again, in yields up to 84%. Okay, and carboxylic acid diversity produced these molecules, again, up to almost quantitative yield. I would say this is quantitative yield in this case. So we have a lot of diversity, and we have good chemical reactivity, which is important. And then she looked at the aldehyde diversity, and in the aldehyde diversity, we also got some very good results. yields of up to 73%. Okay, so what I'm showing you here is the diversity you can achieve here with this UGI reaction using a very important scaffold like isotene. And the ketone is to finalize the ketone. Now, she became interested in looking at a combination of the UGI-Smiles reaction. And the UGI-Smiles reaction is a quite interesting also mixture of multi-component reactions. And when she used her best conditions using these reagents, she got this molecule here, this aromatized isotene, protected isotene, in 24% yield. Okay, so it wasn't brilliant, this result here with the UGI-Smiles reaction, but she incorporated a lot of diversity. She could arylate basically the nitrogen of the amine using the Smiles reaction. Now, the mechanism I won't go into. There's actually a paper was published a few years ago. In fact, it was a French group, EuChemS, I think, developed this UGI-Smiles reaction. So you can look into that later. Now, with respect to the petacesis reaction, in the case of the petacesis reaction, this is, as I mentioned at the beginning of my talk, the petacesis reaction is more recent. So it goes back to 1993. And in fact, it was petacesis was trying to make this type of biologically active molecule, naphthene, okay, naphthene. And he considered using basically a mixture of this vinyl boronic acid, paraformaldehyde, and this benzylamine, methyl-benzylamine compound, heating. And in 30 minutes, it produced this pharmacologically active compound called naphthene. Just to mention some of the disadvantages and advantages of the petacesis reaction, you can use aryl boronic acids. But when you use an aryl boronic acid, there's a pitfall there, a great disadvantage. With an aryl boronic acid, you need to use one that has an ortho OH group in the aryl group, okay? So that's to be based on, no, in that case, you don't need, you need to use an, you need to use an aldehyde component that has an OH group in the ortho position, so salicyl aldehydes, okay? But the reaction with the vinyl boronic acid is much more rapid, and it gives these very interesting products, these allyl amines and also these benzyl amine compounds, which are generally important components in so many biologically active molecules and in APIs. Okay, concerning the mechanism, actually, this is a review. I think this is from Pedro Gais, dates back to 2010. They published a nice review on this work, and what was shown in the review is that in terms of reactivity, the glycol aldehydes are the most reactive, followed by the glycolic acids, and the salicylaldehydes are the least reactive in this case. So the mechanism is basically, again, this addition reaction, formation of an aminium ion, okay? And then what happens is the alpha OH group is very important here. It's critical. It coordinates with the boron. Okay, so we're going back to the boron discussion that we had earlier by Pedro. He's very interesting and nice talk. And then what happens is that we've transfer of the boron, the R5 group, to the imine carbon, forming here the amino alcohol, basically in this case. Okay, and when you look at the salicylaldehydes, the mechanism is almost the same. Form the aminium ion, then you have formation of the 8 complex, okay, and then what you have is transfer of the aryl group, okay, from the boron to the carbon, forming your petacesis adduct in the end. So we published a paper a few years ago. This is the work of Carolina again. So she developed, again, instead of having a nitrogen here in this case, we have a boron here, a boronic ester. Okay, so she incorporated the boronic ester here. So the boronic ester component also contains the isotene unit. Okay, so various salicylaldehydes were used, various amines were used. It's not mentioned here, but we looked at the asymmetric version of the reaction. And the catalyst used here was bienal. So we found that, and I'll show in the next slide, or one of the next slides, the optimization work she did. But bienal was found out to be the best catalyst for the asymmetric version of the potassises. So the reaction is quite moderate in terms of conditions, mild, I mean, 50 degrees, 24 hours. And you can get, she showed 19 examples, yields of up to quantitative, EEs up to quantitative. And also we looked at diastereoselective aspect of the reaction. We got DEs of up to 86%. So with respect to the borylation and the boronic esters, so we've been doing borylations. Well, when I was in Everett, we were doing lots of borylations over the last 10 years. And we've become experts in doing borylation work on arenes, okay? And Carolina showed that she could produce this type of, it's called neopentyl glycolate boronate in yields of up to 76% yield, and using these conditions. And she found that, in fact, the neopentyl glycolate boronate was more stable than the usual picolinate boronate or piconol boronate ester. Okay, so she did some screening work. And let me say again, we have our bienal present here. So that means that we were getting enantioselective reactions. And in the reaction that she tested, we could get EEs, as I mentioned. I think we got an EE, but it's not on this slide, up to 99%. On this slide, the best was 88%, as far as I can see on this one. Okay, and we get very high yields up to quantitative. Now, going back to the catalyst screening, yeah, that's another part of the talk. Okay, so the diastereoselective part. We published a number of papers on doing arylations at the 3 position of the isotene, using rhodium as our catalyst, and also using boronic esters and boronic acids. So we could form this starting material in scalemic form. So it was highly it wasn't an enantiopure, but it was up to 70% or 80% EE in enantiomeric purity. So the idea was to conduct the Ugi reaction, the potassium reaction, using this substrate. So we'd create 2 stereocenters in the molecule. When she looked at the diastereoselectivity of the reaction, we got some good diastereoselectivities. So in one of the cases, we could get a diastereoselectivity of the order of 7 to 93. Now, the absolute configuration, we could never really pinpoint absolutely, because we did some X-ray crystallography, and unfortunately, the X-ray crystallography didn't pan out the way we wanted to. Actually, what we crystallized out was the racemate, and the enantiomerically pure adduct remained in solution. That was quite interesting, but it didn't show any shine any light on the absolute configuration. So basically, what we had to use was the conventional model, to understand the enantioselectivity of the reaction. So the conventional model is basically this one here. So here's your aldehyde or salicylaldehyde formation of the aminium. Then what happens is you introduce your bienal. What happens then is the bienal substitutes the picolinate around the boron. So that introduced the asymmetric induction, okay? And what happens is we believe that what's happening is we're getting our reface addition of the aryl unit of the isotene to the carbon of the aminium, okay, species. And then there is hydrolysis, because we believe there's some water, vestigial water still present, that is responsible for hydrolyzing out for the catalytic cycle, for maintaining the catalytic cycle, okay, and maintaining the quantity of the bienal present in the reaction. And just one more slide that I forgot to mention, we could also scale this up to a 1 gram scale. So she showed nicely that you can also scale up this potassium reaction. So with 1 gram of starting material, boronic ester, and she produced this adduct in 85% yield and an EE of 83%. Removal of the acetal is not so straightforward. You need to use TFA, at room temperature, but still the yield is not quantitative. Okay, so another part of the project, or part of this work involved, and this would involve Pedro Brandão again and Carolina. We looked at another molecule, which is a very photoactive molecule called tryptantrene. Okay, tryptantrene, as you can see in this slide here, has many functions. Not only is it involved in organic catalysis, it's also shows antimicrobial activity, and there's also it also has drug likeness. Okay, so we're coming near to the end here. The synthesis is straightforward: isothene with isotoic anhydride, and the screening reaction to develop the petasis reaction showed that in this case, vinyl again was our best catalyst. Okay, vinyl was the best catalyst, and okay. The EE is not shown here, but we did later some catalyst screening work, and again it was the vinyl that gave the best results. Okay, vinyl gave the best results. Although we did get some good results with diisopropyl tartrate and another diol, which is available from a company called Caratechnics, also gave EEs of up to 60% in this case. So Carolina then did some UGI reactions, not UGI reactions, petasis reactions with this substrate, and also with this substrate, so creating a lot of diversity. And we got some good yields. Some are low yields, but in most cases they're pretty good. Now I must say that as we were using vinyl in this case, all these, although it wasn't tested, all of these products would be not an antimerically pure, but antimerically enriched. However, there wasn't enough time to test each of them individually for their particular EE. Okay, so to finalize my talk, I'm still within time. I've spoken a bit about catalytic UGI reactions with isothene substrates. High chemical diversity has been obvious in all the slides I've presented. I talked about the catalytic UGI SMILES variant, catalytic petasis reactions on isothene and oxindole substrates. Again, I also finished off with some slides on the catalytic petasis reaction on tryptantrene substrates. And we're currently working on the Bucherer-Bergs reaction. So that will be interesting, the next seminars that I give. Okay. And for further reading, we've published a number of reviews on the subject of isothene-based multi-component reactions over the last 3 or 4 years. And of course, I would like to acknowledge my co-workers, as I mentioned, Pedro and Carolina, and Marta Pinheiro from the University of Coimbra, who is also a co-supervisor of Pedro. And these are the financiers, and this is the doctoral program for which Pedro was involved. These are some of the agencies that finance this work. These are some of the group members, okay? And that's, I already showed a picture of Carolina, yeah. I know that Arthur already showed a picture of the universities in Portugal. This was from the EuroJoc from 2013. It just shows the locations of each of our universities. I'd also like to, I know that Arthur already mentioned EZCCAT, but it's going to be in Coimbra in 2025. We're hoping for a great event again, and the Faculty of Pharmacy. And of course, I'd like to thank everyone for their attention, okay? Thank you.
Well done, Anthony. Excellent. The only rearrangement you didn't discuss was the Evra to Coimbra rearrangement.
I didn't, yeah. That's right, yeah.
Super stuff. Again, open for questions from the audience. I don't see any questions coming through in the questions and answers. So Anthony, you've obviously looked at the UGI reaction, and you're right, you've been able to develop a diverse library of isothene-derived compounds. Now, you kind of mentioned a little bit about the biology, but is there any standout property or function from any of the compounds you've made that you'd like to tell us?
Yeah, isothene goes back a long time in my group, and we have, before that, we've been doing, as I said, we've been doing air relations on the tree position of the isothene. So it produces oxindoles, air-related oxindoles. And we also made they're hydroxyoxindoles, basically. We also made amino oxindoles. And generally, they've shown a remarkable butyryl choline esterase activity. So that's involved in cancer. Yeah. So we've a number of publications over the last 10 years on that aspect, butyryl choline esterase inhibition. And it's been found to be selective, not for acetylcholine esterase, but for some reason, butyryl choline esterase. We believe it's because of the isotyne structure itself. Like it interacts in the catalytic site of the enzyme, so it blocks it more easier. We've also got some very good results with for cancer as well. Because I did show in some of the slides that we stuck a triazole appendage. Maybe I can just throw it up here if I think I have a bit of time.
Yep.
It should appear. Okay, can you see it, the slide?
Yep.
Yeah, so it's not that slide, it's another slide. Just wanted to show you. We've put triazole appendages like this one here.
Yes.
And we have here the OH aryl or NH aryl. And when we have a triazole unit, it really interacts really nicely with the butyryl choline esterase active site. So we've got, and not only that, as a cancer as well, we have a publication from a MedChem publication from 2 or 3 years ago. And we've shown some very interesting activity against cancer cell lines. And we're trying to figure out exactly what is the target. We've some idea that it's some kinases or it's post-transcriptional factors or something like that. So, but they have so much biologic activity, isotynes. And I know that my ex-student Pedro and Carolina as well, they are advocates of isotynes. They love them, the isotynes, yeah.
We do a bit of work on oxindoles ourselves, but mainly at the sort of palladium catalyzed decarboxylative or alkylation type strategies. They're certainly an interesting class of compounds.
Interesting class of compounds.
Yep. Any other questions? If not, bang on time. I don't think this has ever happened before in the highlighting series that we've been bang on time. Normally we're over time. We're actually smack on time with our last speaker, and I let Arthur introduce him, but I'll just say I first met Carlos back in 1996 when I organized a young chemist workshop in Dublin. And of course, Carlos, you and I, we would still say we're young chemists, so I have to take a front at someone earlier saying 50 was old, but we'll let that one go just for the moment. And I'll pass over to Arthur to do the more formal introduction of Carlos.
Yeah. So thank you, and thank you, Anthony, for your excellent talk. Carlos, yes, we know from each other for this time too. So we are young at that time, but I think that we can say we're still young at this moment too, and we'll proceed in that way. Well, Carlos has a long history in the organic chemistry in different universities, but now he is in Lisbon in the Faculty of Pharmacy. It's indicated in the program also the research center where is involved. But then today he will speak about the last topic that he will be engaged and publishing on this topic. It's the synthetic transformation of biomass, but today is using flow conditions, and I think we will have. And now Carlos is also the president of the Portuguese of the organic chemistry division of the Portuguese Chemical Society. Carlos, the floor is yours.
Hey, good afternoon. Thank you very much for this introduction. And Pat, I would like to acknowledge the organizing of this event, and Arthur for the efforts during the last years in order to provide more visibility for the Portuguese organic chemistry, and also very involved in our Portuguese Society of Chemistry that I'm really proud. And thank you very much for allowing to come here to present some of the work that we are doing. So as you mentioned in the title, I will provide an overview of some of the work that we have been done on the flow by applying flow. And So when I compare the batch with the flow, in fact, the batch process is the reaction occurs along the time. So when you look for the concentration, for example, for one starting material, the concentration decrease. In case of the flow, at a specific time inside the reaction, we have the same behavior as with the time under batch. So this is a little quite different approach between flow and batch. And flow has some advantages, of course, some disadvantage. And probably some of the future of the flow, such as using lower working volumes and for immobilized catalysts is quite efficient. But I would like to highlight that, in fact, the flow adapts to the setup. The flow setup needs to adapt to the chemistry, to the aim that you want to do. And I think this graphical abstracts from this very good review shows different types, especially of reactors that in fact follows this line. That there is some beneficials of the flow. And I just would like to highlight reactions under extreme conditions, reactions that needs the preparation of dangerous intermediates, and also is quite useful for in case where we have immobilized catalyst due to the higher catalyst substrate ratio, and in case of phytochemistry is quite essential, and also in the electrochemistry. So the first example that I would like to share is regarding the oleuropein, that is a natural compound that is present in the olive leaves in around 1.5% weight by weight, and it contains a glucose part and also a very interesting monoterpene part that is quite rich on functional groups and contains the chiral center and then the hydroterrazole side chain. The oleuropein is already commercialized as a food supplement. So because we wanted to take advantage of the core of monoterpene, Lydia tried to see if you could remove some of these parts, the glucose and also the hydroxytyrosol. And by screening, she found that the amber list was quite efficient. And then we tried the flow, and we just immobilized the amber list inside each EPLC column. And in fact, it worked for 4 cycles. But the substrate was also adsorbed on the amber list, since if you wash, then some product came out. We also tried some efforts with Cassie in order to perform the reaction using the Kuder extract. And in fact, the reaction works under batch condition, but under flow conditions was not competitive with the batch. So the other example that I would like to present is regarding with lupanin. Lupanin is an alkaloid present 1.4% in the lupin beans, the Lupinus albus, and they are used, especially in the Mediterranean region, as a food. But we need to remove the alkaloids because they are quite toxic. And at that time, there was a problem that due to the lack of availability of the spartain, and there was a news in chemical engineering news in 2017. And at that time, and also I just would like to mention that regarding to this process, for each ton of lupin beans is necessary about 10 to 13 tons of water, and also in each cubic meter of ton of wastewater, there is around 3.5 kilograms of lupanin. So it's quite a big amount of lupanin that is really available. For example, here in Lisbon, there is a company that during the summer produce about 7 tons of lupin beans per week, and they export quite a lot. So at that time, with my colleague Nuno Maulid, we developed a process in order to take advantage of the racemic glupanin, because the natural compound from these beans are almost racemic. So we developed a process for the chiral resolution of using tartaric acid, and then the reduction using sodium boron iodide in the presence of iodide. And this is currently under commercialization by the Spartax company, which is a startup company from Nuno and João Seixas. And then using this consortium as a Waterworks consortium, we also developed a process based on membrane technology that allows the isolation of racemic glupanin without the need of high volume of solvent. So with this racemic glupanin in the hands, we, in collaboration with my colleague, the senior researcher Jaime Coelho from the Faculty of Science, close to the Faculty of Pharmacy here. So Raquel Durão explored the possibility to achieve some functionalization using electrosynthesis, and she's screening different conditions, and in fact she was able to achieve an isolated 80%, and did just by isolation with extraction that was by NMR. And also this process under batch conditions, she could run around 9 times the reaction. Then when she washed the electrons, the system recovers again. So then we tried to see if you could perform the flow chemistry, because it will be more important for this process. And in fact, we started with the electrosyn flow from Nika. And in fact, Raquel already, then she optimized different conditions. She was able to achieve around 70% yield. But unfortunately, this process was not reproducible. So due to the design of the reactor and this specific reaction. So then we moved to another setup that is from Asia. And in case of Asia, Raquel Durão, in fact, was achieved the transformation very reproducible. And she tested different concentrations, and also under these conditions, she found that just by tuning the electricity parameters, she was able to achieve the oxidation on the 17th position by insertion of the cyano group, but also using constant current, more drastic conditions, she achieved the further oxidation in 68% yield. And the system is quite robust, as you can see here. Raquel did this study for 5 hours under continuous operation, and you can see that it works under this concentration, more concentrated, and also to achieve the oxidation. So the next example that we explore is regarding to the colophony. Colophony is a quite important industrial product from this country, and it is obtained from the pine rosin, from the type of pines that are grown in this country, and it has many industrial applications. There are quite a lot of industrial sites in this country. And the Portuguese colophony is quite different from the other colophonies because it has a quite high amount of abietic acid and also some dihydroabietic acid. That's why the value of the Portuguese colophony is higher, or at least is different from the other sources. So Inês Martins, also with my colleague Jaime Coelho, she looked for the possibility to achieve the oxidation of the hydroabietic acid. I will present the results of the hydroabietic acid, and also by optimizing the conditions under batch and flow, she was able to achieve the allylic oxidation of the methyl ester, the hydroabietic acid, the abietic acid itself, and quite important, use some commercial colophone that is enriched in the hydrobiatic acid, and INEJ was able to achieve from this quite complex mixture the oxidation either in flow and in batch. And the system is also quite robust, and we tested for the methyl ester and for the hydrobiatic acid. So another part that I would like to describe is regarding to some furan building blocks that are prepared from carbohydrates, and we have been working for some time on the preparation of HMF. And in fact, around 10 years ago, we described the process that was performed in flow, and we also applied this to flow chemistry only for educational purpose. And in fact, this was the first paper in chemical education regarding to one process under flow conditions for a synthetic approach, not just for analytical. And taking advantage of furfural in the HMF, we explore different types of chemistry, and more recently, Rafael Gomes explored the preparation, and with Juliana and Bruno, they explore the preparation of some furan derivatives derived from ketene, and also the diazole reaction that allows the formation of quite crowded products, and also the formation of more functionalized cyclopentaenones, since we also work on these cyclopentaenones. And we also explore some of these transformations under flow conditions that I'll also explain in more detail. One first example that I would like to present is regarding the functionalization of the ethyl furan dicarboxylate, that is a quite important building block. And Carlos Monteiro and João, they found that using CalB, they could achieve the chemoselective amidation. And then then João played a little bit with the solvent, and he observed that also Xylene could work, because in collaboration with my colleague Giovanni Poli from Sorbonne University, João found that he could achieve the CH in certain specific indisposition derived by the amide bond. And then João just did an enzymatic transformation. The product goes to the schlenk that contains the reagents and the catalyst for the CH insertion. Then he heated the system as a normal reaction in the schlenk without any ORCAP. He was able to achieve the two-step transformation, and in which one is under flow conditions. So the other transformation that I would like to highlight is the formation of diaminocyclopentaenones from furfural. And Beatty reported a very, very efficient catalyst for this system, and in which the reaction occurs via formation of Steinhals and then electrocyclization. And after Rob Beatty, there have been many catalyst systems for this transformation. So Raphael looked for the possibility to achieve this reaction under flow conditions, and he tested different catalysts. And he observed that in fact was possible to immobilize copper in silica, and then in collaboration with my colleague Andrea Peixoto from the Faculty of Science from University of Porto. So they gave us several catalysts in which this one was quite efficient for this transformation, including the case in which we have also some groups in this position. And also Raphael and Lydia Cavaca, they also did the reaction under flow and compare with the best catalyst that we found for this transformation. And you can see that in this case, the flow provide better yields than in case of batch. And then, because we wanted to see if the flow was quite efficient, we selected this diaminocyclopentaenone containing the two diallyl groups. The reason is because Rob Beatty described the best, the shortest total synthesis of this quite important compound that I will describe in more detail. And so you use this compound as a model. And in fact, Raphael found that the flow was quite robust. It worked for the maximum time that we tested was 300 minutes. But also with other catalysts, it works until 200 minutes. There are some benefits from considering the green matrix for this process. So at that time, Lydia looked for some of the reactivity of these diamine cyclopentanones, and she found that when you hydrogenate this bond, there occurs a quite easy elimination. And in fact, by screening the bases, just potassium carbonate was quite efficient. So we could perform the hydrogenation using the H³ mini, and then perform the elimination with potassium carbonate. So then Lydia with Raphael, they combine the two processes in which we have the first flow process for the aminocyclopentanone. Then we need to inject ethanol in order to have the right flow for the H³ mini, then perform the second reaction in the flow, and then in the beaker we have just carbonate, potassium carbonate for the final elimination. And she achieved a production around 1 gram per liter per hour. So during the studies, Raphael found that in fact our catalyst, the catalyst that we described, copper triflate, was quite fast in water, and just after 2 minutes at room temperature, it was able to form the aminol. And the aminol is with Juliana, they screening the substrate cope, and here you can see that also works for fuflol and the HMF. And because the reaction was quite new, so we looked at the possibility to may describe this as a possible protective group. So Raphael and Juliana, they looked for the condition for the protection, and also the possibility of some reaction that mimics the potential use of the aminols as a protective group. And with this knowledge, so Raphael saw about the possibility to use this reaction and to apply for educational purpose. So he optimized the process, and these are the picture that the students did in the laboratory. We had around 30 groups of students in which they immobilized just copper sulfate and methanol. They put just in a normal column chromatography, and then just did a flow reaction in the column in which the flow rate was adjusted just by the stopper as we do the chromatography. And then it just evaporated the solvent, and this is the product that the students obtained. So this is really a basic flow process that allows the students to understand the concepts and that are behind the flow system. So regarding to the gelastatin, this is a quite active, bioactive compound, has been isolated from the sea sponge. It has a quite high level of toxicity in the range with a variety of human cancer cells in the range of nanomolar scale. Is already known the mode of action in which has been already reported the X-ray structure of the gelastatin binding to the protein, and also has been reported 15 total syntheses in which 6 are acyl and 9 are chiral. So here just shows the range of cells that has been tested the cytotoxicity. And they are, as I mentioned, in the range of nanomolar activity. And also is also known for different derivatives that the positions that I put here in red are absolutely crucial for the activity. So from the system that we have, we may be able to perform the total synthesis of this molecule. And I highlight here three total syntheses, two are chiral, in which one of the steps to create the chiral synthesis is based on enzymatic resolution. And then again, the beta total synthesis, that is the shortest one, in which he used this diaminocyclopentanone containing the bis dialylamine, as I mentioned, in which this very efficient synthesis as a challenge step that involves the protection of the two groups and then needs a differentiation of the free amino groups. So Raphael saw about the possibility to apply more the total synthesis of raw beta, but instead of having the same groups, protective groups on the amine, you may be able to achieve the synthesis of the mixed diaminocyclopentanone. And in fact, he screened quite a lot the conditions, and he was able to do that. And in this case, that was unfortunately not the best example. He achieved 64% yield. And he also tried some attempts for the flow, but unfortunately in this case, the flow was not competitive with the batch. So we need to live with 64% for the first step. And then by adapting the total synthesis from Rob, and then he was in fact able to achieve the total synthesis in 10 steps, 25%. He had some problems that was necessary to reduce the carbonyl, the ketone, and then oxidize again. But this process allows now the selective, the protection of this dialyl group, insertion of this part, then the protection again of the other one, insertion of the urea part, and then follow the total synthesis. But this process, since we were unable to achieve the chiral cyclopentanone, we saw about the possibility to perform also the asymmetric version. And since Filipe Ciopa, that is a senior researcher here, and with João, they also studied this transformation in later Milan under flow conditions. So we had already experience for the transformation of pyridinium salts to this quite variable bicyclic aziridine. So we saw about the possibility to use this 5-member ring, which contains already five chiral centers, in order to insert selectivity each group, and then follow more or less the raw total synthesis. So to do that, we need the starting material, and we selected the allyl group, but unfortunately the photochemical reaction is not very efficient. We had other flow system, but it was necessary to build another flow setup. And I built this setup, in fact, at home, that is consisted for 26 UV lamps that is inside this tube, and then we insert the reactor that is in fact corresponds to 12 reactors that are 12 quartz tubes in each independent line by using a peristaltic pump. And by doing that, Milan was able to produce around 130 milligrams per hour, and this we have now enough starting material. And with this starting material, then Milan studied the resolution of this bicyclic aziridine, and by screening some enzymes was in fact able to achieve this resolution with Cal-B. But unfortunately, under batch conditions, because the aziridine is not very stable, occurs the ring opening. So then she's screening different conditions, and she found that triethylamine using triethylamine, the CALB still work and minimize this undesirable reaction. But she was able to achieve 50% yield, which is good, is the maximum, but the yield was around 80%. So then we looked for the possibility to perform this flow that may avoid the side reaction. And then using just a small reactor where she put 140 milligrams in HPLC column, she optimized the conditions, and in fact, she was able to achieve 30% yield and with 93% in enantiomeric excess as expected. So you reduce the yield, we increase the yield of the product. And also the system is quite robust. So she worked for 26 cycles. The yield was always very good, and there was a slight erosion on the yield. But then when we determined the absolute configuration, we were quite disappointed because, in fact, the product provide the wrong configuration in order to achieve the total synthesis. So we need to go back again and now find conditions in which you can produce the starting alcohol in high yield. But since we were using flow, was more easy. So we just screened the residence time, and she was able to achieve 48% yield with 95% enantiomeric excess. So with these conditions in hand, then you need to scale up the process. So the way how we did, in fact, the scale-up approach in which you just increase the reactor. So we did 10 times the previous scale, for now with 1.4 grams of CALB, and for this we put inside the Teflon tube and just cut the tube in the end, we put inside the chamber, and then we ran the reaction to collect the product at low temperature. And with these conditions, Milan could give 260 milligrams to Joinville with 98% enantiomeric excess, that correspond to 46% yield. And with this starting enantiomeric pure by cyclic aziridine, then Joinville just applied the total synths that he already developed before with the racemic bicyclic aziridine and also containing the butyl group. And the key points, I will not discuss this in more detail because it's not the aim of this presentation, but I just would like to highlight that this aziridine is absolutely crucial because he was able to ring opening with the azide, and then he could insert here the urea, and then later by reduction he could insert the other part and then perform that was following the synthetic group from raw baited, but in fact was quite different some steps because due to some features of these different molecules. And then he was able in fact to achieve the total synthesis of enantiopure angelostatin in 12 steps, 4% overall yield, and just one protective group, and with two steps on the flow conditions. And I would like to highlight that without the flow process, we will not be able to achieve this asymmetric synthesis because we could not get enough starting material. So with this process, then, as I mentioned before, is already known that these positions are absolutely essential for the activity, but was not known the effect of this NH bond. So since we have João already synthesized the racemic angelostatin and also the CARO1, and this analog and the other with the butyl, then Cassia tested the biological activity with these three cell lines and compared with the racemic angelostatin, and as you can see, in fact, this NH bond is absolutely crucial, is also important for the activity. So now we can say probably that this is also an essential position. So from the examples that I mentioned here, and also in line with the literature, so flow conditions can be an advantage approach for the transformation of variable synthetic building blocks and many other transformation as well. And the combination of more efficient synthetic methodologies implemented in our flow conditions may provide additional benefits to more sustainable chemistry. So I would like to acknowledge you and the team and the colleagues that has been involved during the last years, and also the very important financial support. And thank you very, very much for being here and for the possibility to present the work. Thank you.
Carlos, thank you very much. What a superb way to finish up. That was a superb lecture. Congratulations, super stuff.
Thank you.
Again, open now for questions from the audience. I mean, that was a real tour de force in, I would say, modern synthetic organic chemistry, the use of biomass. I mean, we're all interested in the circular bioeconomy, but not many of us are actually really doing it. So that's very impressive. But also the combination of flow chemistry, photochemistry, electrochemistry, you know, really, I really enjoyed that. Congratulations. So again, open now for, I'm sure there'll be many from the audience or from our fellow panelists. Paula.
I can ask, it's only a very simple question. Carlos has a lot of experience in flow chemistry, and do you have any advice for someone that wants to start? What type of conditions, reactions that can be used, or is there something that we should avoid?
Well, I think from the examples that I provided, I think this first slide is probably very important for when we start. I think the main point is we need to adapt the flow setup to the chemistry or to the purpose. And I think I'll show you a very simple purpose for education that allows to teach the concept. But in some cases, you need to use more elaborated systems, and there are now some commercial setups like from Vaportec and from cities. So I think we can either try to order this, to have these flow setups, the commercial ones that we don't have at the moment, but we can also create the setups for the specific purpose. I think I cannot give a general answer because of that, and also this, as I mentioned, this graphical abstract exactly shows the issue of flow. We need to adapt to the system.
Great, thanks, Paula. Maybe Carlos, I'll ask you a question. One of the valorization of furfural was to go to make cyclopentenones, but in particular cyclopentenones that have a trans 1,2-diamino setup. So, I mean, cyclohexyldiamine is one of the go-to chiral diamines that people look at, and stilbene diamine would be perhaps the other go-to chiral backbone that people look at. But I'm not aware of anyone that has looked at, and you'd have to resolve them, but it's still an interesting potential application of those cyclopentenones. Do you know, has anyone looked at that?
For the chiral version?
Yeah.
Yeah, if we look for the mechanism here, in fact, we tried quite a lot with Juliana in order to achieve the symmetric version. Yeah. And the main problem is, and I think I may go also to another slide, but the main problem of this transformation is that this process is reversible. Okay. So we need to perform this transformation quite fast and then maybe trap the cyclopentanone, but the chiral version has not been reported so far.
But even to have the racemic mixture and resolve it, that would still be of use.
Yeah, yes, but we need to trap. Because if we resolve, then it racemizes, and you know that because you already observed that. So we could resolve In fact, we saw about this, but then we need to trap quite instantaneously the cyclopentanone, otherwise it will racemize.
Great. Okay. We have one question in from an anonymous attendee. Great talk. Speaking of creating the setups, do you think that it is useful to 3D print flow systems for testing reactions, or is it better to buy different setups?
No, I think 3D prints are really very, very good. And you may find there are several groups that work on that, because we can create the reactor on demand. So depends of the chemistry, you can create the flow reactor as you wish. And this is very, very good approach, yes.
Good. Okay, I don't see any further questions. So maybe if you stop sharing your screen, so please, Carlos.
Oh, sorry.
So just some final comments. It's been a long day now. It feels a long time ago, since 9 o'clock this morning, or 10 o'clock if you're Central European time. But I think we really have had some highlights from organic chemistry in Portugal, and it's been, for me, a very interesting occasion. And I'd like to thank, first of all, Arthur for organizing everything locally in terms of the speakers, but really to thank all the speakers for superb talks, starting off with Arthur in terms of some background on the history of organic chemistry. And I found the exponential rise in publications very interesting. I think many of the people around this table, are responsible for a lot of those rise in publications. So well done to all of you. And with interesting talks all around. Teresa starting off with spirocyclic beta-lactams and pyridine-fused chlorines, a lot of photodynamic therapy. That seemed to be a bit of continuity with lots of other talks as well. PDT certainly turned up. Platinum complexes. We then moved over to Augusta, who talked to us about diketopyrols, I should say. I learned more about the Ferrari red, and even the date of the revolution in Portugal was thrown in there just for good luck. So that was interesting as well. Bit of PDT is in there also. So color chemistry was very obvious going through lots of the other talks as well, because of course we had where is the one now on wine? That was Victor. So lots of anthocyanins, etc. So like colors, and it was interesting how tuning the color was so important in terms of the flavillene compounds. So lots of stuff going on there. Pedro, lots of chemical biology, antibody drug conjugates, and really the functional boron containing complexes and how you can manipulate them, and really they're an important new conjugate when you look at what goes on in terms of chemical biology. So then we moved on to Paula, who talked a lot about synthesis of aminals and how certain aminals, that cyclic system was particularly tricky in terms of stopping it racemizing, but when it doesn't racemize, it's a really important part of synthetic organic chemistry, and lots of interesting mechanistic studies to try to pin down exactly what was going on there. And then we moved on to Anthony, who talked to us a lot about rearrangements and multi-component reactions, the UGI reaction, the UGI-Smiles reaction. I didn't know about the Smiles reactions. That was something new. And even one rearrangement was new to me as well. So lots of things to learn through all of that, as well as the potassium reaction and developing the highly enantioselective version of that. So well done, Anthony, and all of that. And we finished up, as we just heard, with Carlos giving us a tour de force in biomass to compounds that you can increase value of, and then being able to use those as starting materials through a combination of batch chemistry, but more particularly flow chemistry, photo flow chemistry, electrosynthesis flow chemistry. So in terms of a highlight and a sort of snapshot of organic chemistry in Portugal, I think it's been a really superb morning, and I really want to thank you all for your contributions. And it's been recorded, and you can send it to all your friends once we bring the YouTube web link available to you. So again, thank you to everyone involved, and a date for your diary. The next Highlighting Organic Chemistry series will feature the United Kingdom, and that will be on Friday, the 24th of November. And Will Unsworth, our Division of Organic Chemistry Committee member, is the organizer for that. So again, have a good day. If it comes to sport, in soccer terms, we've a very poor soccer team. Portugal has got a very good soccer team. In terms of rugby, Portugal has not got a very good rugby team. We have a very good rugby team in Ireland.
We need to win tomorrow.
And we hope very much to beat the Springboks tomorrow evening at 8 o'clock. I'm sure, Anthony, you'll be wearing your Irish jersey.
Oh, I will indeed, yeah.
So will I. And hopefully we can beat them and go all the way and finally win the World Cup. That's our plan, and we look forward to that. So enjoy the rest of the day, and thank you, and bye-bye from Dublin.
Okay.
Bye-bye. Bye-bye.
Thanks
Thank
a
you
lot,
very
Pat,
much.
and everybody.
Bye-bye.
See you, everybody. Bye.
All right.